Yeah.
Welcome, everyone. This webinar is already set up to be recorded, so we'll be collecting all your faces and voices as you speak. We're just asking people to enter their name and affiliation into the chat right now. I will be closing it up in a little bit, but we'll just wait a few more minutes. Thanks for being here.
The CDC says you can take either. I remember from the first time I was
Good afternoon, everyone. Thanks for being here on a Friday afternoon. My name is Pam DeBona. I'm the Executive Director of Massachusetts Bay's National Estuary Partnership, or MassBays. We had 100 people registered for this webinar, which is a testament to the importance of this issue and the critical need to respond. So we'll just be sure to introduce yourself in the chat with your name and affiliation, and maybe a note about your interest in this topic. I'll be closing the chat at about 2:10, so we won't be distracted by side conversations. And instead, we're asking that people use the Q&A button at the bottom of your screen to pose questions as we go along. Let's see. Oh, there is an option to upvote or recommend others' questions by clicking on the thumbs up icon, and Zoom moves that question up in the queue. So oops, hang on a second. Our agenda consists of 3 parts. In the first part, the first 3 presentations are designed to bring us all to a common understanding of the conditions in Cape Cod Bay over the past few summers. And at 3 o'clock, we'll shift to presentations that explore factors that may be contributing to the documented conditions with regard to low dissolved oxygen, especially nutrient inputs. The second part of the webinar, beginning after the break, will be with start with a case study, and then includes a panel discussion with the speakers and additional representatives of stakeholders and agencies. And during this discussion, we'll do our best to set out next steps, informed by both the science and the requirements for more data, research, or other supports. In the third section of the forum, we'll invite questions and suggestions from the participants, especially comments on factors we might have missed or additional responses that should be considered. Up until that point, participants will be muted and you won't be able to use your cameras. We sprung for the webinar version today. Please use the Q&A function. As I said, you can enter your question using the little button at the bottom, and then once you pop up the Q&A window, you can push on the little thumbs up button, and that way we know that it's a popular question, and we'll do our best to get to it sooner than others. So just to let you all know, following the forum, we'll be compiling notes from the discussion for our Science and Technical Advisory Subcommittee of MassBays to review and to produce recommendations for action. So let's get started, shall we? And I'll ask Cressida Vela, our staff scientist, to introduce the speakers in turn.
Hi, everyone. Thank you again for joining us. I will be actually introducing the speakers as they go through their presentations. So first up, we have Dr. Judy Peterson from MIT Sea Grant. She's also the chair of MWRA's OMSUP, which is the Outfall Monitoring Science Advisory Panel. And she will be starting this off by talking briefly about OMSUP's role and the interest in prompting this forum. I'll pass it on to you, Judy.
Okay, thank you. Is this the right one? I hope so.
Yes.
It is, yeah. And I just need to get down to the bottom here to, whoops.
Yeah, the little icon.
Up, actually. Okay, so I'm not able to see it.
You have to go to the bottom.
Yeah, I know, and I'm not able to see it on my screen is the problem.
Okay.
Let's see if I can get this out of, where am I? Did I lose it again?
We see your screen, but not the presentation, maybe.
Yeah, so I will.
You can unshare and kind of start it over.
Okay, be able to do it. Yep. Yeah, it's the bottom that's the problem for me right now. Hang on. I knew this would happen. I have a very small screen and I can't get rid of the information. Oh, there we go. All right, got it. Sorry.
Perfect.
Yes. Okay, so what I'm supposed to do today is give you an introduction to and the context for the forum. I want to thank Pam and Cressida and the MassBays program for putting this together. I think it's really important. So what I'm going to do, I have a lot of words on these slides and I'm not going to read the slides. I'm just going to basically try to highlight what I think is important to sort of lay out the issues. So as we all know, back in the 1980s, Boston Harbor was a mess and the MWRA was established to build a wastewater treatment plant that was going to address the issues. And in the process of doing that, they were given the permit to put the outfall 9 miles out into Massachusetts Bay. It's caused a lot of heartrending concern on the part of individuals and NGOs from the Cape and from the North Shore. And so as things moved forward, we decided that, and we were able to talk to MEPA to suggest that we have a monitoring program that would say, allow us to evaluate the effects on MassBay. And so the Outfall Monitoring Task Force advised MWRA and their consultants, and the task force was composed of scientists, agency folks, NGOs, and developed their monitoring plan. By 1998, this was sort of institutionalized in the permit, the National and Pollutant
Discharge,
NPDES, National Pollutant
discharge elimination system.
Thank you. Discharge and elimination system. I do know it. Anyway, they appointed a scientific panel, which is the Outfall Monitoring Science Advisory Panel, and 2 advisory committees, the Interagency Advisory Committee and the Public Information Advisory Committee. And the monitoring plan itself had basically 4 sort of major issues. Is it safe to swim? Is it safe to eat the seafood? Do you maintain the aesthetics? And the big one, of course, was the do no harm to the ecosystem. In addition to the monitoring plan, contingency plans around the monitoring and the effluent were developed. And these provided what we call caution threshold levels and warning threshold levels. And they were set up to provide a basis for evaluating if things were starting to go in the wrong direction. In other words, if you were seeing an increase in, say, cadmium, you would be able to use these thresholds to look at it. And certainly this applies to nitrogen and to a lot of the other issues that we are dealing with. The Outfall Monitoring Science Advisory Panel was set up to represent a variety of backgrounds. So we have physical, chemical, and biological oceanographers. We have biologists. We have people who are experts in looking at microbiological organisms and low DO to marine mammals. And we have statisticians, people who are good with not only the physical oceanography, but also modeling. So it's a really good and very distinguished group of people, present company accepted. The data on the contingency plan that we look at are things that are integrated. So look at mussels contamination, for example. So they're pulling in contaminants from the water, dissolved oxygen, which of course is a combination of things like temperature, salinity, depth, and so forth, and algae, as well as the very specific things like pH, fecal coliforms, toxicity, and nitrogen, and so forth. So our responsibilities that are to report to the 2 agencies that are responsible for the NPDES permits, US EPA and Mass Department of Environmental Protection. We are expected to conduct peer reviews of the MWRA's monitoring reports, which they give us every November. We evaluate the monitoring data and provide any insights or implications on that data to the agencies. We also advise on any modifications to the monitoring plan. We meet regularly with the 2 agencies and discuss any issues and to make sure that if there were issues, that there is a scientific approach to addressing those on the part of MWRA. And we also play an important role in looking at the warning levels and the caution levels as they are occasionally exceeded or met or what have you, and give again advice to the agencies on these. So and the one thing I always like to remind people is the MWRA is not the only wastewater treatment plant in Massachusetts. We have a number of others around the Bay Area itself, as well as what is coming in from the Merrimack. And there are some larger ones, and I don't know if it's the Massachusetts ones or the ones in New Hampshire, but there are some that are still not really doing a very good job of dealing with their wastewater. So the issue was we had noticed through the years that the nitrogen levels were gradually creeping up, and you can begin to see that. This slide is from MWRA's 2020 report for the 2019 data. And as you can see, I just don't know how to get, there's a black line right in front of me. So there are three things. There are the bars represent the nitrogen. It's either ammonia, which is the gray, blue, or grayish color. The dark blue is all nitrogen, not accounted for as either that or the light blue, which is nitrates and nitrites. And the lines represent the caution level, which is red, the blue, which is the warning level, and one that was calculated by modeling the 120%. What happens if you increase your nitrogen by 120%? Would you expect to find problems? And what you're seeing is that basically in 2019, MWRA's nitrogen levels increased and did, in fact, go over the caution level. So we started paying attention to it. As I understand, if I remember correctly, I don't have the data, that in 2020 it did not exceed the caution level, and of course we still don't have the 2021 data. So this, of course, raises some concern. We are worried about this. We know that nitrogen, and I know you'll be getting much more of this from Tracy and other speakers, that nitrogen is responsible for increasing phytoplankton, and phytoplankton that is abundant will decrease and fall to the floor, and so on and so forth. And as a result, if you do not have a way of mixing that, you will get layers that begin to build up and use up the oxygen, and so you have a hypoxic event, which is in fact what is showing up in Cape Cod Bay. The issue of the increased nitrogen really triggered a discussion with OMSAP members, and it served to foster a really deeper understanding on the causes of hypoxia, which I think we all know something about, but of course, we're talking about an ecosystem, and it's not easy to pinpoint any one specific thing. I'm happy to say that that in addition to the hypoxic events that we're seeing in Cape Cod Bay, in the quarterly report this year, the bottom waters, the oxygen saturation of bottom waters in both, particularly in Stellwagen Bank, are below the warning levels, or at the warning levels, and even their DO is fairly low. So, we are seeing changes in MassBay that are probably not just related to increased nitrogen, but some of the other things that are going on. For example, our sea surface temperatures are changing. We know that other nutrients are probably also changing. There are a lot of other climate-related events that are occurring. And these are some of the things that the OMSAP group discussed and raised as issues, and we were pleased to have this group come together and give us some of the information that they're seeing, so that when we go back and meet again, we can begin to talk about this in terms of some of the things that are going on and that people have also been looking into and can give us some insights. And finally, we do have the MassBays, MWRA's Bays eutrophication model, and its predictive capabilities can be used for things like assessing excessive nitrogen. As I said, in the past we've used it to double the nitrogen level and see what kind of an effect it would have on nitrogen in the bay, and quadruple it. And because so much of the nitrogen is coming in from the north and from the Gulf of Maine, it makes it very difficult to say what these increasing changes from MWRA are adding to the whole system and to the impacts that we're seeing. So it's a really interesting problem. It's a difficult problem to kind of tease apart, but I'm thankful that we have such good people here. So some of my take-home messages from this are three things. One is, I honestly believe that MWRA has been one of the stellar agencies that we've worked with in terms of trying to do good science and trying to make sure that we are not polluting our bays and our waters. I think that the setup of the ambient monitoring plans and the contingency plans has been a good model for being able to set some sort of level against which you can, a measuring stick as it were, against which you can say, okay, things are changing, and here, this is a point at which we want to know, is it changing, continuing to change, or was it just a one-off, or are we going to be looking at further problems down the road? And some of the changes have not been all bad. We saw an increase in diversity in the sediment, or in the benthic communities, and people said, what is that? How do we deal with this? And I personally think it's a good thing, because we want lots of diversity out there and in fact, it was a sign that the sediments were much cleaner than they had been. And then finally, well, there are other, certainly many other important studies, research studies that have gone on in Mass Bay and are continuing, and other monitoring programs, the Center for Coastal Studies, for example, and Stellbank Bank, among others, that help us understand the MassBays program. I really do think also that the long-term monitoring by MWRA is a powerful data set, and it's something that has been underutilized by many of our folks. So from our perspective, I think I'm speaking hopefully for AMSAP, that we are looking forward to getting this information and being able to come back, take a look at these increasing nitrogen that we're seeing from MWRA, and start to try to understand what's going on in the larger sense. And with that, I thank you. Pam, feel that I don't need to have any questions, because I think it's important to go on to the other people. So thank you.
Thank you very much, Judy. That was really an excellent introduction. And we wanted to start with Judy, because this the impetus for this forum came by after an OMSEP meeting. But EPA felt that they wanted to ask MassBays to do this, because the questions are actually even bigger than MWRA. MWRA is also one of the many other sources into Mass Bay and Cape Cod Bay, and we're here to kind of examine other possibilities that could be affecting our ecosystem, or rather these showing these DO changes, etc. So as we move ahead and we hear from the scientists, then we'll come to questions on how, based on what we heard today, where would we move forward? So we're really looking forward to that. So thank you, Judy, for introducing. And now the next speaker is Dr. Tracy Walsh from BMF, Mass Division of Marine Fisheries. She's the senior marine fisheries biologist there, working on invertebrate fisheries, including lobsters. So Tracy will be presenting the work that has been done to date by the Cape Cod study fleet, which in collaboration with some fishermen as well as researchers. And we are really looking forward to some of their initial findings over the last couple of years. So take it, Tracy.
All right, I think I'm making progress here.
Yes.
Good. All right.
Perfect. Yeah.
Let me move that. All right. So thank you to the organizers for asking me to speak at this. I appreciate that. And I am going to talk about sort of how we first noticed these hypoxia conditions and how that led to the initiation of this Cape Cod Bay study fleet. This is work with Division of Marine Fisheries, also the Lobster Foundation of Massachusetts, and the Mass Lobstermen's Association. So, oops, hang on, I'm having trouble advancing. This worked, I promise it worked.
Yeah, sometimes, Tracy, are you trying the back and forward buttons?
I'm trying the up and down.
Okay, try the mouse, click with the mouse. Yeah.
Oh, all right.
Yes,
The roller on
there
the
you
mouse. Go. Okay. All right. So we have to go back in time to September of 2019 to set the stage here. And during this month, late in the month, we got phone calls from the Mass Lobstermen's Association reporting multiple reports of dead lobsters. There were about 8 fishermen who were reporting dead lobsters in their traps, anywhere from several dozen to hundreds of dead lobsters. So way, way, way more than normal. And they were also seeing dead crabs and finfish in their traps here. This all sort of took place around the 20th to the 24th of September. We did receive a report from a dragger, a mobile gear fisherman, sort of in the middle of the bay, who was reporting larger than normal proportions of dead scallops in his catch. And all of these reports seem to be focused around the southern portion of Cape Cod Bay, roughly the area east of the Cape Cod Canal over to the Barnstable Harbor entrance. And depth ranges were around 30 to 80 feet. Everything seemed to be focused in that depth range. So DMF initiated a pretty broad scale investigation immediately into figuring out what was the potential cause here. And as we got on the water and really started trying to check things out with the assistance of the Center for Coastal Studies, what we found was some pretty severe hypoxia in portions of the bay. Hypoxia essentially just means that there's not enough oxygen in the water. And what we were seeing is, if you look at this cluster of points down here, if you can see my cursor, there's a cluster of red points down here on the southwestern portion of the bay. Those were all areas where we were seeing values less than 2 milligrams per liter of DO. And that's low. Several of these locations were actually less than 1 milligram per liter, which is really low. And then we had this broader spread of points with just 2 to 4 milligrams per liter, which is also, we would consider that hypoxic. These hypoxic areas were concentrated very close to the bottom. The upper portions of the water column were fine. The DO levels were perfectly fine up there. It was just near the bottom. And this hypoxia, based on our sampling, persisted for at least a week, but based on the reports of dead lobsters that we were getting, we know it lasted a little bit longer than a week. And for context, the levels that we detected when we were out there to measure are definitely lethal to lobsters. After an exposure of about 48 hours at around 59, 60 degrees temperature. So Judy went over this briefly, but I'll do it again just in case we need a refresher. So how does hypoxia form? Essentially, nutrients come into the system, and these nutrients act as food for things like phytoplankton, and they result in increasing organic material or algal blooms. The algae then dies, and it's going to sink to the bottom, and it's going to decompose. That decomposition actually uses up oxygen in those bottom waters. And then when you have a really stratified water column, which essentially just means the surface waters are much warmer and less dense than the bottom waters, which are colder and more dense. So there's a lot of separation that prevents the surface waters from mixing down and bringing that oxygen back down to the bottom. So over time, the bottom waters are depleted of oxygen, and that forms hypoxia. Most mobile animals can actually detect changing conditions, and they're going to try to move out of the way to avoid conditions that they don't want to be in. Unfortunately, if those animals are stuck in a trap or they are not mobile, so something like a clam, they can't get away. And so they're going to be exposed for that over a prolonged period of time. It will kill them. So our concern with this was really, all right, we saw this in 2019. Is this a real anomaly, or do we need to worry about Cape Cod Bay becoming a seasonal dead zone here? As we all know, have heard, the Gulf of Maine is warming more rapidly than many other parts of the world's oceans. And the problem here is that warmer waters actually increase the potential for hypoxic conditions to develop. Warmer waters hold less oxygen. They further stratify the water column, so they make the difference between the surface waters and the bottom waters that much more. The warmer waters tend to increase the biological productivity, and they tend to increase the sediment oxygen demand. So in order to answer this question, we really need a better understanding of what kind of processes are driving this oxygen depletion in Cape Cod Bay. And I'll give a little bit of a teaser. Malcolm's going to talk about this in more detail. But one of the things that we can look at to see if 2019 was really an anomaly is chlorophyll. Chlorophyll represents phytoplankton, essentially algae. And if you look on the top graph up here, this is sort of the average chlorophyll from 2010 to 2018. The bottom graph is 2019. And you can see it's pretty different. So 2019 really stood out in terms of the average chlorophyll for the year. So really, in order to address these questions and really figure out what's going on here, we needed to figure out some new ways and some additional monitoring efforts that we wanted to put into place for 2020. And this was done sort of in consultation, the lobster fleet in Cape Cod Bay, when we met with them sort of after the DO event had sort of cleared, we talked with them and they were really interested in getting involved and in trying to help figure out what's going on and, being essentially really involved in the process. And that led to the development of this Cape Cod Bay study fleet. So with the fishermen in the area and the Lobster Foundation of Massachusetts, they were able to get some funding from the Massachusetts Climate Change Resilience in Fisheries and Aquaculture Grant Program. That was in early 2020. And what we did with this is they were able to purchase a system that consists of a deck box and a data logger, which you can see in the upper right here. And the data logger collects data on dissolved oxygen and temperature. And you attach this thing to the traps. So they go overboard with the traps and they log the data. It was recording data every 15 minutes. And then when the fishermen haul the gear back, the data logger can talk to this deck box via a Bluetooth signal, dump the data, reset itself, and so it's ready to go when the guys are ready to put the traps back in the water. So it's an automated process. It's really easy, and it gets really nice long-term continuous data on the bottom. The deck box will then dump all of the data to a server on shore, and that's where DMF staff came in and we sort of managed and handled the data. So the 2020 fleet, if you can see on the chart down here on the bottom, this is sort of the coverage that we had. So we have pretty good coverage in the southwestern portion of the bay and some coverage up in the northeastern portion of the bay. And just to very briefly go through a little bit of what we saw in 2020, I'm going to talk about the loggers that came from this cluster of points down here in purple near the Sagamore Beach area. So these graphs, there's one for each of 5 loggers here. And you can see the dissolved oxygen is in blue, and then the temperature data is presented in centigrade, and it's in orange, but I'm going to focus on the DO. And the first thing you can see here is we have a very clear decline that all 5 loggers showed us that took place from around the middle of August through about the first half of September in 2020. Following that, we had this brief little uptick, so an improvement in mid-September or so. And then again, most of the loggers picked up a decline into early October, followed by another improvement. And then we have one final little downward blip in late October that most of the loggers consistently picks up. So what we get with these data are really high resolution in situ measurements that really give us a good idea of the trends of what's going on, and we can look at this in detail over time, which is really nice. The 2020 study fleet was a really great success, and because of that, we went ahead and continued the program into 2021. So these are the monitoring locations for this season. Monitors went into the water roughly in July and are just starting to come out of the water now. And one of the big improvements we were able to make this year is on sort of the data retrieval end. So last year, DMF staff, myself and others, were sort of manually dealing with retrieving the data, processing it, and getting it into some sort of format where we could try to figure out what it was telling us. This year, we worked with our Department of Fish and Game GIS staff, and they have developed this data dashboard for us that automates a lot of the work that we had to do previously. So it automates the data retrieval, the processing, gives us this nice visual, and gives us this spaghetti string mess on the bottom here that looks like a mess, but is actually super useful. So what you're looking at here is each of these points represents an average over a 6-hour time frame. So you know, from midnight to 6 a.m., 6 a.m. to noon, each of those time periods has a data point at these locations. And then the data points are color coded based on our system over here. So anything in red is what we would consider to be severely hypoxic. That's less than 2 milligrams. And it's basically a stoplight system. So green and blue are good. We consider that normal. And then you can see we have a fair amount of yellows and some oranges on this graph. The nice thing I can see from this, even though it does look a little bit messy, I don't see any red, which is really good. We did not see anything dip into severely hypoxic in the 2021 season. We do, however, have a couple of instances of loggers dipping into the orange. And if you look at the logger specific data, which is what's down here in the graph below, you can see there's a couple loggers that dip into this color shading of the orange along here and, you know, over time. So this is only showing data from mid-August through about last week. But you can see in mid-August, we have a logger that spent some time down here. And then in late September, we have one here. So this is generally difficult to interpret, but I can see that we did have this period here of declining DO, and it was pretty consistent throughout a lot of these loggers. And if I want more details, this data dashboard tool lets me look at specific loggers or specific time periods. So if I take a look just at the specific logger that was up in the northeast portion of the bay, I can see its location here, and for a little while it was over here. And on the bottom here, I can see this graphic of what it was detecting over time. I've got another logger down here, and this is in the area where we normally expect for the hypoxic conditions to set up based on what happened in 2019 and 2020. And we can see that from the middle of August or so, it was a little bit low. We had some big improvements, dips down, it goes back up, and then it spends some time relatively consistently low here and continues to decline a little bit into late September. And then all of a sudden conditions change and it increased pretty dramatically, pretty quickly. So these data loggers really do give us a really nice high resolution image of what's going on over time. One of the things that we're working on for next season to try to develop is something that will allow the public to view these data. So we're working on a publicly accessible web application where, so this screenshot here shows, for example, loggers from the last 7 days. And you can see that the locations and the app will essentially allow someone to either tap or click on a specific location and get more detailed information about it. So the time span, again, this is a 6-hour average. So this is from 6 a.m. to noon. Here's your average DO of 7.2, which is fine, it's good. And then you can click through here, you can click through all the observations. So you can see that there's multiple observations at this location. So that's what we're working on for the future, to be able to not only let the study fleet and the other lobster fishermen in the area have a tool to look at recent conditions, but any user who would have an interest in the area. And I just wanted to, thank the study fleet. I think that their interest and their participation in this has really allowed for this this kind of data collection to develop. The guys have been great, and they certainly are telling me that they're looking at the duck boxes and they're picking up information and learning from what they can see just on the boat and with the equipment that they have. And again, this was supported with the Lobster Foundation and the Mass Lobstermen's Association, with our departmental Fish and Game GIS folks. Lowell Instruments, who are the manufacturers of the equipment, have provided excellent technical services. They've been great to work with. And the GIS development was funded by a sea grant from the American Lobster Research Initiative. And if I have time, I'd be happy to take some questions.
Thank you. Thank you, Tracy. That was a really, really good. I'm always excited to hear about the study. I think there was a lot of work done, right? Jumped quickly into this because it was kind of what's considered a little bit of an emergency too. There is one question for you from Jeffrey Rosen. How were the sensors calibrated?
That would be a technical question that the manufacturer would have to answer. I don't know, but we do, they get calibrated before they go out for the beginning of the season, and at the end of the season, they come in and they go back to Lowell, and the technical folks at Lowell Instruments go through a postseason calibration. And if I remember correctly from last season's, he was really happy with the performance. And there was very little drift over time, negligible essentially, that they didn't worry about what was going on, that the sensors were getting less accurate over time.
Thank you. There's also a couple of, we'll take a couple more questions now before we proceed to the next. Another question you have is, can you overlay on the logger data maps the broader bay currents, municipal outflow sites, and locations of ecologically sensitive locations?
I would imagine that will be totally possible. We're still sort of in the, what I would consider the learning phases of getting the data spatially managed. The loggers collect a lot of data. So now that we're doing better with handling the data incoming and the processing, I think that the next step would be making it available to the folks who would be able to do things like that. So I don't have access to those data sets, but we can certainly share the study fleet data with folks who would be interested in taking it that step further and figuring out what current patterns or anything that it's associated with.
Okay. Okay, great. And in order to keep on time, Pam reminded me here, sorry, but the panelists and the speakers, you have the ability to see the questions. So any questions directed like that, you can also answer by typing into the Q&A as well. And everybody will be able to see the questions and the answers at that time. And then, of course, there will be time for more during the panel discussion and subsequent Q&A period. So thank you, Tracy. That was awesome. We now go next to a group of speakers. So we have Dr. Malcolm Scally from Woods Hole Oceanographic Institution. He's an oceanographer there, and he and Amy Costa from Center for Coastal Studies, as well as Rocky, I think he will be chiming in as well. They're going to have a presentation that will look a little bit at the physical oceanographic conditions in Cape Cod Bay, and also at long-term data on DO and temperature from various monitoring programs that have been happening over the years, particularly from the Center for Coastal Studies, but also more recently from data gathered as part of this project that we have here. So I pass it on to you, Malcolm. You can see your screen.
Okay, can you hear me?
Yes, I can hear you.
All right, I'm going to go without video. I'm hoping my internet holds up to this presentation.
Great, thanks.
Yeah, and if I start breaking up, please let me know and I can go back and try to repeat.
Certainly.
So what I'm going to do is I'm going to present the results of a project that's in collaboration with Rocky Geyer here at Hui, Tracy, who you just heard from, Amy Costa and Owen Nichols at Center for Coastal Studies, and David Borkman also contributed as well. And this is something that was funded through the NOAA Sea Grant American Lobster Initiative, as well as some additional funding that I received through the National Science Foundation for a rapid award. And so really the goal is just to present sort of a retrospective analysis of what happened in 2019 and 2020 and see if we can come up with some hypotheses about what drove the hypoxic conditions that occurred in those two summers. So this is just a map of data that Tracy just presented, but it's looking at the minimum bottom dissolved oxygen that was recorded from sort of a Southern Cape Cod Bay wide survey that was conducted in 2019. There was 3 discrete cruises on September 26th, which is the stars, September 30th, which are the squares, and October the 1st, which are the circles. And then the color bar is just the bottom oxygen in milligrams per liter. And so what you see sort of centered between the sort of 10 and 20 meter isobath is this sort of broad region of hypoxia that was really what alerted us to the conditions in Cape Cod Bay and that this was becoming a problem. And so this is what we observed in 2019. We obtained some money to do more detailed surveys in 2020. And we went out on a regular basis to measure both the vertical profile of dissolved oxygen as well as through spatial maps. And so we did a series of cruises, and this is what we saw in 2020. We first became aware that another low oxygen event was occurring in sort of late August. We got out and we did sort of one across shelf transect, and that's what I'm showing in this upper left panel. So these are, again, just the bottom dissolved oxygen that we recorded, and they're color coded. And then the size of the circle, the bigger circles are for the lower oxygen values. And so we went out in October or in August in response to some reports that the oxygen was getting low. And indeed, we saw another hypoxic event developing with a similar sort of a cross-shelf distribution of low oxygen as we saw in the previous year. We started regular surveys. We went out on September the 3rd. We did a more comprehensive survey on the 3rd, and that's in this upper right-hand panel. And we saw this broad band of low dissolved oxygen consistent with the 2019 distribution. Again, this was centered in relatively shallow water between the sort of 20 meter isobath and the nearshore zone. We went out on September the 10th. Again, the region maybe got a little bit smaller, but again, there's this sort of continuous band of low dissolved oxygen water that we found in September 10th. We went on the 16th. It had moved offshore and maybe expanded somewhat. And then we had a big wind event. And by September the 24th, essentially the oxygen had returned to sort of normal conditions. We had one final survey on October the 2nd. And again, we saw no hypoxic conditions. We saw slightly lower oxygen at the offshore locations than we did on September the 24th, but no hypoxic conditions existed on October the 2nd. So I just want to, this is stuff that Tracy just talked about, but I just want to show how the 2020 hypoxic event was captured by the study fleet data. So this is a compilation of all of the loggers, all the bottom loggers, and the oxygen data that was collected by the lobster fleet in 2020. The map in the top, the blue circles indicate where the data was collected. I've color coded all the data that was less than 3 milligrams per liter in red. And those loggers that recorded the low oxygen are shown on the map as red stars or red asterisk symbols. So basically, the data that we presented from our surveys is largely consistent with what was captured by the study fleet data. The nice thing about the study fleet data is it's a much more continuous time series. And we basically see that as we moved into August, we see this decline in oxygen in sort of mid to late August, with the onset of hypoxia occurring in late August. And it sort of persisted till roughly around September the 20th. And then there was a large wind event that sort of mixed away the oxygen and returned the bottom to normoxic conditions. So I just want to highlight the role that these late fall or late summer, early fall wind events play in modulating the oxygen. This is the survey from October 1st, 2019 in the upper panel. This is data that I've already showed, but this is showing the relatively large region of hypoxic bottom water in early October 2019. In response to that, we went out about a week later, and that was after a relatively intense northeast wind event. So we get starting in the fall, we start getting these northeasterly wind events, kind of like what we had over the last several days here. And these are very effective at mixing the water column combined with the surface cooling. And so while we had relatively widespread hypoxia on October 1st, when we went out on the 7th after this strong downwelling event, which is winds from the north, we basically mixed away the hypoxic bottom water. I want to call your attention to the fact that a couple weeks earlier, we had a relatively strong wind event from the south, and I'm calling that an upwelling wind event. That did not mix away the conditions. And so it's really these late fall storms that tend to be most productive at mixing away this hypoxic bottom water. We had a similar phenomena occur in 2020. So I'm comparing in this upper plot, the bottom oxygen on September the 16th, where we had relatively widespread bottom hypoxia. And then if you look about 8 days later, after one of these large northeaster wind events, we see that the bottom had largely been returned to normal oxygen values. And so this, in the bottom panel, I'm showing the north-south component of the wind. The red is highlighting that strong northeast wind condition that mixed the water column and effectively ended hypoxia in 2020. So in both 2019 and 2020, we had persistent low oxygen conditions, both of which were mixed away by strong wind events from the northeast, these sort of early fall storms. So we also conducted detailed across-shelf surveys, and I just want to present some of the data from these across-shelf surveys. This is an example of that data. So on the x-axis, this is degrees north. So this is a transect that we sampled going from the south to the north. The y-axis is depth. And in the upper panel, I'm just showing the two-dimensional distribution of dissolved oxygen. These red colors near the bottom, these represent that hypoxic or anoxic water. The blue colors represent the well-oxygenated surface waters. These white contours are representing the temperature gradient that we observed. So we have sort of relatively warm near surface temperature around 20 degrees, with much cooler, say 12 degrees C bottom water near the bottom. One of the interesting things that we note is that this hypoxic water is really limited to that bottom layer below this intense thermal gradient. So this thermal gradient is very resistant to vertical mixing. We can mix oxygen down from the surface to the depths of that thermocline, but we're not able to mix oxygen across that interface. And as a result, the degradation of organic material below that thermocline is what depletes this lower water of oxygen. Some other interesting things to note about the oxygen distribution is it tends to be lowest where that bottom layer is thinnest. And as we move offshore and that bottom layer gets thicker, the oxygen goes up. And that can be understood in terms of the fact that what's driving the oxygen down is the consumption of organic material near the bottom. And when the layer is thinner, you can more quickly deplete that layer of oxygen than when the layer is thicker. And so we see the most intense hypoxia is occurring where this thermal gradient is very, very close to the bottom and where the bottom layer is very, very, very thin. The other thing to note is that when we look at the chlorophyll distribution, is that we see very intense subsurface chlorophyll fluorescence indicative of a bloom of a phytoplankton that's occurring at depth within that thermocline. So the surface waters by late summer and fall, early fall, are largely depleted of nutrients. The bottom waters below the thermocline tend to have high nutrient concentration. And so what we hypothesize is going on is that there's a bloom of a phytoplankton within the thermocline, and that has to be a species that is adapted to low light conditions, and it's maintaining its position in the thermocline where it can have access to the subsurface nutrients. And so these are the detailed across shelf, or one example of the detailed across shelf surveys. We did a number of these, and we saw that there were pretty dramatic responses to the wind forcing. So I just want to highlight the difference between the response to a wind event from the south, which is in the upper panel, to a wind event from the north. So we did a bunch of surveys. Our third survey was on September 10th, and that occurred after a period of relatively weak winds from the south. So in this picture, the wind is blowing from the south, which is from the left to the right, or offshore, upward away from the shoreline of Cape Cod. When we have these upwelling winds, that drives the surface waters offshore, and we have the bottom waters being driven onshore. So that drives colder water near the bottom onshore, and that advects this low oxygen water onshore. And if it goes far enough onshore, it can get in shallow enough water where it can mix, and that can alleviate these low oxygen conditions. The other thing to note is that when it blows from the south and offshore, the temperature gradient in the water column becomes more spread out or more diffuse. And I want to contrast that with the bottom panel, which was 6 days later after two relatively energetic wind events from the north. So this is blowing from the north, it's blowing towards the southern coastline of Cape Cod Bay. And in these cross-shelf transects, that's a wind that's blowing from the right to the left. So that's driving this oxygenated water onshore. There's downwelling at the coast, and that's pushing this temperature gradient closer to the bottom. And so that has two effects. One is it's intensifying the gradient near the bottom, and it's also making that lower layer thinner. And as I've already mentioned, the thickness or thinness of that bottom layer, we think plays a big role in controlling the near bed oxygen dynamics. And so this bottom layer between the 10th and the 16th got much, much thinner, and we see that the oxygen levels dropped, and the overall region that was hypoxic expanded to a much larger area on the September 16th survey. The other thing to note is that in the September 16th survey, we have this well-mixed upper water column, a strong thermal gradient at depth, very close to the bottom. And within that thermocline or thermal gradient near the bed, we have, again, high chlorophyll fluorescence. So these winds from the north are intensifying the near bed temperature gradient. They're making the bottom layer thinner, which we think might be contributing to the occurrence of hypoxia. And we also think that's favoring blooms of species that are capable of existing at depth and utilizing deep nutrients. So I just want to highlight the difference between a vertical profile taken at the same location, separated by 6 days. The gray line is what the temperature profile looked like after the winds had been blowing from the south, or what I'm calling upwelling conditions. And the black line is the temperature profile that was taken 6 days later after the winds had been blowing more from the northeast, what I'm calling downwelling conditions. So even though the overall gradient from the top to the bottom is gotten bigger due to the upwelling, the downwelling has really intensified the gradient near the seabed. And that can be seen in the panel below, where I'm just plotting the vertical profile of the temperature gradient. So this is the vertical gradient in temperature. And you can see that after this downwelling event on September 16th, that gradient is almost a factor of 10 larger. And that temperature gradient corresponds with a much thinner bottom layer. And if we look in the upper right-hand panel, we can see that that was associated with much lower dissolved oxygen. So this is just one location, and things are advecting back and forth, but it does highlight the fact that these very thin bottom layers are more conducive to low oxygen conditions. They're also more, they're more favorable for these subsurface chlorophyll blooms of phytoplankton at depth that seem to be associated with a strong thermal gradient near the seafloor. So I just want to summarize briefly what the data that we collected showed us. So in both 2019 and 2020, the bottom waters in southern Cape Cod Bay became hypoxic. We're not sure how long the hypoxic conditions persisted in 2019, but we think in 2020, they lasted for about 20 days. In both years, a strong nor'easter effectively ended the hypoxia. This occurred earlier in 2020 than in 2019. So we think the timing of these sort of big fall storms may play some role in how long the hypoxia persists. The hypoxic bottom waters were observed and associated with a strong near-bed thermal stratification, with the lowest oxygen values occurring where the bottom layer was thinnest. We saw very high subsurface chlorophyll fluorescence, and that was present within the thermocline and just below it, suggesting that the organisms, the algae that's living there, was there because of its proximity to the bottom nutrients. This hypoxic water advects both on and offshore in response to north and south winds. Winds from the south tend to drive the bottom waters onshore, and then the winds from the north tend to drive the bottom waters offshore. And then finally, even though these strong nor'easter events ended the hypoxia in both 2019 and 20, we think that moderate winds from the north and northeast, when there's still strong thermal gradients, tend to intensify the near bed stratification. And this reduces the thickness of the bottom layer, and that may favor both lower dissolved oxygen and also higher chlorophyll fluorescence. So I just have a few more slides, and I really want to just focus in on what we think was different about 2019 and 2020, and why we think hypoxia occurred in these two summers. So to do this, I'm going to present an analysis of historical data that was collected in Cape Cod Bay. I'm going to show some data from the NDBC Boston buoy, which is this square in the upper left of my map. That's data that goes back to, that should say 1984. I'm sorry, that's a typo, but we have data at the Boston buoy going back to 1984 to the present. MDNF has bottom temperature loggers in Cape Cod Bay. I'm going to show some bottom temperature from this triangle location. That's a location that's known as the wreck of the Mars. And so we have nearly continuous temperature at the bottom here at this triangle location. I'm also going to show data that was collected by the Center for Coastal Studies. They go out and do monthly surveys in Cape Cod Bay, and these lettered and numbered stations on the map are where CCS makes their monthly surveys. They measure a whole suite of water quality and water column parameters, including salinity, temperature, dissolved oxygen. But what I'm going to focus on are their vertical profiles of chlorophyll, as well as their bottom inorganic nutrient measurements. And then finally, the MWRA has been making monthly surveys and water quality data at 2 stations that are relevant to this study. F1, which is in the red on the map, and then F2, which is also located in as a red indication on the map. And what I'm going to use from that data is they've been doing phytoplankton enumeration and identification, and that goes back to 1991. And this is the analysis that David Borkman has contributed. So I just want to show all of the vertical chlorophyll profiles that were collected by the Center for Coastal Studies going back to 2011. And I want to show all 8 stations. And so these are the 8 stations in this panel. Each panel is one of the stations. The thin gray lines represent all of the data collected in September prior to 2019. The black circles are the 2019 September cruise data, and the red triangles are the 2020 September data. And the key message here is that there was anomalously high chlorophyll at all of these stations in September in 2019 relative and 2020 relative to the previous 8 years. That's particularly evident at stations 8M and 9N, which are these in the lower box here, where we see the chlorophyll values exceeding 50 milligrams per liter at 8M and getting up to 100 milligrams per liter at 9N in 2020. So these are extremely high values, and they're more than an order of magnitude higher than anything that was observed prior to 2019. So we see this anomalously high chlorophyll, and it's almost always located in these different stations as a subsurface chlorophyll maximum. So this is not surface chlorophyll, but this is chlorophyll at depth. And this is actually pretty deep if you think about how much light is getting down to, say, 30 meters depth. So these are deep chlorophyll maxima that are observed pretty deep in the water column. So I just want to take that data and I want to sort of try to do a more aggregated analysis of it. In the top panel, I'm showing the depth integrated chlorophyll averaged over the summer and averaged over all 8 of those stations. So in the top panel, this dashed gray line represents the 2011 to 2018 mean. And you can clearly see that 2019 and 2020 had much higher depth integrated chlorophyll. It's more than a factor of 3 higher. So if you take that chlorophyll and you convert it into carbon, and you assume that all that carbon was respired in the bottom layer below the thermocline, that would represent somewhere between a 3 to 4 milligram per liter decrease in oxygen. So we think that this increase in chlorophyll fluorescence was more than enough to drive the oxygen from, say, 4 milligrams per liter down to about 1 milligram per liter. So we think that this anomalously high chlorophyll is really what was providing the biomass that resulted in the hypoxic conditions in 2019 and 2020. CCS also collects bottom inorganic nitrogen. So this is all of the forms of inorganic nitrogen in the bottom waters. And this was estimated, the total integrated nutrient content of that bottom layer below the thermocline. And what we see is that there were ample nutrients in 2011 to 2018, but consistent with this large increase in chlorophyll fluorescence in 2019 and 2020, the bottom nutrients were significantly depleted. And that suggests that there was an organism present that was capable of utilizing those deep nutrients. And that's consistent with these deep chlorophyll maxima that we saw in their profile data. So there was something different that happened in 2019 and 2020 that allowed these nutrients to be utilized, leading to this large bloom of phytoplankton. And we think that phytoplankton is what's responsible for the depletion of oxygen. So one of the things that has come out of the long-term MWRA phytoplankton monitoring is the fact that a new species of phytoplankton was detected in 2017. So prior to 2020, an organism known as Karenia micomotoi had never been detected in any of the MWRA sampling locations. It was first detected in 2017, and then at very low levels in 2018, but we saw a big jump in 2019 and 20. In both surface samples collected in 2019 and 2020, the number of Karenia cells was above 75,000 cells per liter. We actually collected one subsurface sample from 2020, and the analysis that David Borkman ran on that sample suggested that there were more than 700,000 cells per liter in that deep sample in 2020. And in that sample, 70% of the phytoplankton that was identified was Karenia. So we have this occurrence of a new phytoplankton species that occurred, that the timing of which is consistent with this high chlorophyll fluorescence that we were observing. So I'm sure David will get into this in a little more detail, but I just want to give a couple of details about this organism Karenia mikamotoi. It's a planktonic dinoflagellate species, and it's responsible for harmful algal blooms in coastal areas worldwide. It's ichthyotoxic. It's lethal to a wide range of fish, shellfish, and other invertebrates. And it's often observed to form very, very intense blooms, and it's been linked to hypoxia in a number of other environments. It grows over a wide range of temperatures, but its optimal range for blooms is thought to be somewhere between 20 and 24 degrees C. It grows well in low light conditions, and it's particularly well suited to exploit low light environments, such as the deep thermocline where we observe high chlorophyll fluorescence. Unlike diatoms, it can vertically migrate. It can move up to 2.2 meters an hour, and it's often observed to form highly concentrated layers within the pycnocline that are typically very, very thin, less than a meter thick in vertical thickness. Again, consistent with our observations. And then finally, it has strong allelopathy, suggesting, which basically means other things don't like to eat it. And so it's very good at becoming the dominant bloom species. So we think that Karenia may have been the species that's responsible for the high chlorophyll fluorescence that we observe. And we think that the biomass of that high fluorescence is what might have driven the oxygen to hypoxic conditions. So I just want to give a quick overview of the physical conditions in Cape Cod Bay. In the upper left, I'm showing the summer temperature at the Boston buoy. This is data going back to 1984 through the present. And you can see that there's this steadily increasing trend in surface water temperatures, with 2019 and 2020 being two of the warmest summers on record. Water temperatures in this area have increased by about 3 degrees Celsius in about 35 years, which is an extremely high rate of surface warming. That surface warming is, the surface waters are increasing faster than the bottom waters, at least from our analysis of the wreck of the Mars bottom data, suggesting that the thermal stratification has gone up. And again, 2019 and 20 were two of the most stratified years. If we look at the number of days where the surface water temperature is above 20 degrees, that's the lower right-hand plot. We see that in the sort of late 80s and early 90s, we would get about, say, 5 days a year on average where the temperature was greater than 20 degrees Celsius. But as we move into sort of 2015 and onward, we see that we're getting over 40 days of greater than 20 degrees surface water at the Boston buoy. So this is consistent with perhaps a thermal threshold being crossed. And then finally, in addition to these large changes in wind or in temperature, we see some pretty significant changes in the summer winds over Cape Cod Bay and the greater Gulf of Maine. This is an analysis in the bottom left panel of the Boston buoy. And what I've done is I've simply figured out the percentage of the time during the summer that the wind blows greater than 15 knots and what percentage of the time during the summer that occurs. And the black circles are wind events where the wind is blowing from the northeast. The gray triangles are wind blowing from the southwest. And what this shows is that over the last 30 years, we have started to get more and more wind events, sort of moderate to strong wind events, where the wind is blowing from the northeast as opposed to the southwest. The stronger winds from the southwest have gone down, and the stronger winds from the northeast have increased. And so, in the mid 80s, we'd expect maybe 20% of the strong wind events were from the northeast. Now we would expect maybe 50% or more of the strong wind events are from the northeast. And so this has big implications for the thermal structure that I was talking about earlier in the presentation. So I'm going to go ahead and conclude with some hypotheses that we've developed based on these observations. So we hypothesize that this emergence of Karenia mikamotoi is consistent with a physical environment that has undergone significant changes, including dramatic warming, increased stratification, and significant shifts in the summer winds. We think the changes in the physical environment have allowed this new species to effectively utilize these deep reservoir of nutrients And that's really allowed it to become established in Cape Cod Bay. We're also hypothesizing that this new species is responsible for the large late summer blooms that we observed in 2019 and 2020. And we think the biomass from these blooms, combined with stronger near-bed stratification, has resulted in hypoxia in 2019 and 2020. And so the changes in the physical conditions in Cape Cod Bay, which we think are really warmer surface waters, more stratification, but also dramatically different winds, we think that that's an environment that's more physically susceptible to hypoxia because it has strong stratification very close to the seabed, which allows the waters to become hypoxic more quickly. We also think that favors subsurface phytoplankton production by motile species such as Carenium micomotoi. So those are sort of our basic hypotheses about why we think 2019 and 2020 developed hypoxia. As Tracy pointed out, we did not have similar events this summer, 2021. Oxygen got low, but it never got the hypoxic conditions. And we never saw really intense subsurface phytoplankton blooms in our data. And so that's an analysis that's still ongoing, but we do have data from this past year. So I'll go ahead and stop there, and I'm happy to answer any questions.
Sure. Thank you very much, Malcolm. That was really interesting and very informative. So it will form a good basis for our discussion later. As we said before, we'll try to address Q&As for now. If you can answer any questions that are for you in the Q&A section. In the meantime, I believe next up is Amy Costa. She has a brief presentation on water quality. Amy.
Yes. Can you see my screen now?
Yes, thank you very much. We're running a little bit late, so we're trying to catch up a little bit. I apologize for some of the timing.
No, I'll be pretty quick.
Oh no, yeah. Take your time. I'm not saying for in general, just so the audience knows. That's fine.
So this is just, I just wanted to go into a little bit more detail about some other monitoring that's been going on in addition to what has been done for dissolved oxygen. This is a map that is pulled directly from our website of all the stations that we monitor at the Center for Coastal Studies for water quality. And on the right are the different parameters that we measure at each of the stations. And so from a first glance, it looks like we have really good coverage of the water surrounding Cape Cod. But if you consider the area that we're talking about today, you can see that there's a pretty significant gap there. But that being said, that, you know, as Malcolm showed in his presentation, the historical data from our offshore stations is still very useful in putting into context what has been going on in recent years. But what I'm going to talk about a little bit today is some of the analysis that we've been doing on long-term trends in the data that we've been collecting at our stations in Cape Cod. So this is a map of the locations of the stations that I included in the trend analysis. So there's about 70 stations located along the shoreline of Cape Cod, in addition to our 8 offshore stations in Cape Cod Bay. On the right, you see the percentage of stations that are showing a trend. The gray indicates that there's no trend, the blue indicates a decreasing trend, and the red indicates an increasing trend. So the top figure just shows the percentage of the nearshore stations. The bottom 2 figures show the percentage of the offshore stations at the surface and at the bottom. So this was brought up because some of the people have, there's been some concern that nitrogen loading from the Cape could be a factor that's contributing to the formation of these hypoxic areas. So that's why I wanted to look a little bit at our long-term trends to see if, in fact, we do see anything of interest. So first, looking at dissolved oxygen, less than a third of the nearshore stations are showing a decline or showing any trend in dissolved oxygen. The ones that are showing a trend, most of them are showing a decline. But what's interesting is that the stations closest to our study area are actually showing an increase in dissolved oxygen. I should also say that these 3 stations in Barnstable Harbor, the data are not collected by the Center for Coastal Studies, but they're collected by the town of Barnstable as part of the MEP program, but they just gave me the data to look at. The The offshore stations are a little bit more consistent with all of the surface data, showing a significant decline in dissolved oxygen, or a significant declining trend in dissolved oxygen. At depth, we also see a significant decline in dissolved oxygen in the bottom waters at the majority, at 2/3 of the stations that we monitor. If you look at chlorophyll A, you see that there are about 40% of our nearshore stations are showing an increasing trend in chlorophyll. Offshore stations, oh, and also again, it's these stations over here that you would expect be the most influential or influenced by the dissolved oxygen hypoxic area. And a lot of these aren't showing any trend at all. The offshore stations are consistently showing an increasing trend in both the surface waters and at depth in chlorophyll. So the story for nutrients is not as clear cut as I was hoping it would be. It's kind of difficult to tease out what's really going on, but for looking at trends in total nitrogen, only about 25% of the nearshore stations are showing an increase in total nitrogen, or an increasing trend in total nitrogen. And again, the areas, the locations of the stations near the hypoxic area are for the most part not showing any trend at all. In the offshore stations, we have a couple stations in the surface that are showing an increasing trend in total nitrogen. And at depth, we aren't seeing any increase in total nitrogen. For dissolved inorganic nitrogen, nitrate, nitrite, it's similar to total nitrogen. There's not that much, about 30% of the nearshore stations are showing an increasing trend. None of the surface waters in Cape Cod Bay, the stations that we monitor, none of those are showing any kind of trend. And in the bottom waters, only one station in the northwest corner of Cape Cod Bay is showing a significant increasing trend in nitrate. Ammonium is a little bit more interesting, but a little bit confusing too, because it wasn't what I expected at all. Very few of our nearshore stations are showing any kind of trend. The offshore stations, both at surface and at depth, are showing a significant declining trend in ammonium. So I guess it's really difficult to make sense of what's going on with nutrients, especially with the high variability along the shoreline of the nutrients. But I guess the take-home message is that our data suggests that nutrient loading from Cape Cod does not appear to have that much of a significant or a direct impact on the formation of this hypoxic zone. But these in the offshore waters, these significant trends of increasing chlorophyll, decreasing dissolved oxygen, and decreasing ammonium should, that's the most interesting to me, and it's probably deserves for further investigation. And that's all I have.
Thank you, Amy. A lot to think about and much food for thought here. And again, this will be really good for once we come to the panel discussion to have this information. I mean, Center for Coastal Studies has been collecting all these data for so many years. Having these long-term trends is really helpful. So the next part, now we're going to look a little bit quickly into some of the sources that we think might have a role to play. So first up is Dan Codiga. He has a short presentation from, Dan is from MWRA, and he will be presenting some of the work related to total nitrogen and the MWRA effluent. Take it from there, Dan.
Okay, hi everybody. Can you see me and hear me?
Yes.
Okay, and my slides showing?
Yeah.
Okay, good. All right, so my name is Dan Codiga. I'm with the Massachusetts Water Resources Authority. And I put together some slides that talk about our MWRA effluent and nitrogen and how they could be connected to Cape Cod Bay. And so this is a collection of slides that review results in previously published documents and reports. And I'm going to try to catch us up time-wise today. I'm going to maybe skip a few slides and see if I can do that. So don't worry, all this material is in the file that has been posted in the collection of resources that were prepared and put on Google Drive. So if you see something here and you'd like to learn more about it, you can always find it there. Something to keep in mind about all these studies, they're generally not focused on Cape Cod Bay. They're focused on Massachusetts Bay. But I'll be trying to highlight the connections and relationships potentially to what's happening in Cape Cod Bay. And the general structure of the talk is I'm going to present our observations first, and then I'll move on to some of our modeling results. And at the very end, I have a couple slides that are from just this past summer. So just to bring everybody back to the 1990s and when the outfall planning was occurring, to just remind you that the outfall is located 9.5 miles or 15 kilometers offshore, and it began operating there in 2000. And that location is at least 37 kilometers from Cape Cod Bay, the very northernmost edge of Cape Cod Bay. It's an important number to keep in mind, 37 kilometers. So during the planning process, there was a lot of concern about potential eutrophication and the idea that excess algae from the effluent could elevate plankton growth, and that decay of algae would consume oxygen. That's why the outfall was located 15 kilometers offshore in water that's 30 meters deep. And this location does stratify each year from about April or May till about roughly October. And this has the effect of trapping the effluent from the outfall at depth below the picnic line, which is generally deeper than where the well-lit areas that plankton tend to grow best at. And we think that helps reduce the possibility of eutrophication. And I just also point out that before and after 2000, the outfall moved from the location here on Deer Island to out offshore. And the idea that these nutrients from our effluent are making it down to Cape Cod Bay changed when the outfall moved offshore. We think that since then, the amount of our nutrients that could reach Cape Cod Bay would be less because of this deep water location where the mixing is better. So this is a plot of our observed ammonium from the outfall. Ammonium has been proven to be the one parameter that our effluent most affects. And so it's a good way to see the signature of our effluent. If you look at the map in the upper right here, first, you see this red transect line. It's about 50 kilometers long. And so the data presented in this graph are showing the distance along that transect in this in the direction from left to right here on this direction of each of these slices. And these slices are 5 slices, horizontal slices at various depths. The uppermost one is the surface, the lower one is the seafloor. And so that's the 2 dimensions here, vertically depth and then distance downward to the right. And the final dimension is the year in time. And so this is a picture of averaged ammonium concentrations during that stratified period. And you can see the outfall startup in 2000 here. You can see increases in ammonium that occur along that transect at the location of the outfall. So the brighter colors here are the relatively higher ammonium. You can see that during the stratified period, they are most concentrated, the high values are concentrated mostly at depth beneath the picnic line as planned. If you then look at the other time of year when the picnic line is not present, unstratified conditions from December to March, you see that the ammonium is mostly found up near the surface as expected. So that's kind of as planned. And the increase is, it's important to note the distance here. It's not as clear on this plot as some others that we've made, but the distance of the increased ammonium is located within about 10 to 20 kilometers of the outfall. I'm going to skip this slide in the interest of time, but this slide shows some individual one-day survey results, which also confirm the same characteristics that I just described on a seasonal mean average. So the point here is that on a shorter timescales, there's a lot of variability in the currents and the winds, and the effluent is moving around. And so on any given particular day, it might extend further in one direction or the other, but we do this every year. And we see that it generally extends at most 10 to 20 kilometers from the outfall. In this plot, we're looking at long-term averages over a 10-year period, 2011 to 2019. I guess that's 9 years. And the vertical mean conditions. And I want to highlight here over the lower left, These are stations that are organized by distance from the outfall. And you can see very clearly the blue line is the ammonium. That's what I was just showing you in the previous slides. Ammonium is elevated, and that drops off as soon as you get to about 8 to 10 kilometers away. So on the long-term mean basis, the ammonium is elevated only within about 10 kilometers of the outfall. And of course, the DIN, the dissolved inorganic nitrogen, which includes ammonium, also is elevated, as well as the total nitrogen. That's this black curve here. That's also elevated, mainly due to the ammonium. And so this is something we feel like we really understand well where the signature of the effluent is occurring in terms of the nitrogen species. Interestingly, if you do the same calculation, same plot, same averaging for chlorophyll, which would be some indication that the nutrients are causing elevated algal growth, you don't really see that signature. You can't really detect the presence of the effluent in the chlorophyll here. This is the middle panel, the green dots. And similarly, in particulate organic carbon, another parameter we might expect to be elevated if there were elevated phytoplankton. So these are some important conclusions from the long-term mean analysis. So I think I'm going to skip this slide in the interest of time. This slide summarizes the dilution of the effluent from both models and observations. And the bottom line is that there's strong evidence that by the time the effluent might reach Cape Cod Bay, it's diluted by at least 400. This is a model result from an early model result from the 1990s, where a budget for the mass balance of total nitrogen was done across Mass Bay and Cape Cod Bay. And the results of this budget were that the MWRA contribution of total nitrogen is about 3%. And this is not a particularly precise value, but the point is that more than 90% of the nitrogen is coming and going in the system from the offshore source. And so this was an early result, and I'll show you next. I'm not going to go through the details here just in the interest of time. I'll show you next a more modern update to that result, which is from a recently updated version of the model. And it gives a very similar kind of result with the loads from effluent and rivers and atmosphere being something like 1/12 of the loads that are coming in from offshore, from the offshore boundary. So the very early model and the more modern model are telling us the same thing, in effect. It's a very strongly boundary controlled system in terms of the nutrients. And this is another model result. This is a comparison of the conditions at the outfall station and at a station in Cape Cod Bay for 3 parameters: the seafloor dissolved inorganic nitrogen, the surface chlorophyll, and the seafloor oxygen. So if we were looking for, and we do this because we're looking for potential eutrophication effects. And what we see here is that if you run the model in a scenario mode and you change only one thing, which is the amount of nitrogen coming out with the effluent, the concentration of nitrogen in the effluent, you can increase it by a factor of 10%. You go from black to red, or by a factor of 2, going from black to blue. You see that at the station over the outfall, you do see a linear increase in the seafloor DIN. However, at that same site, you don't see much of an effect at all in chlorophyll, and certainly not in oxygen. At the same time, down in Cape Cod Bay, there's really no influence at all, even for a doubling of the effluent concentration. Okay, these are my last couple slides. This is, I wanted to put the Cape Cod Bay hypoxia that we've learned about in the previous presentations in context. So this is a plot of September measurements of oxygen at the bottom, both in Mass Bay and in Cape Cod Bay. And the point here is that up in Mass Bay, there's no hypoxia. The oxygen conditions are quite good in sharp contrast to what's happening in Cape Cod Bay so far away. So again, no hypoxia in Massachusetts Bay. And I'll show you the next slide here, results from the Stillwagon Basin Station, where we had a recent exceedance of our contingency plan. And that slide was shown earlier by Judy, and just want to briefly describe what's happening here. The shaded colors here indicate the range of conditions over 28 years, the median as well as the min to max range. And then the year 2021 is in black, superposed. And then we have these threshold values as well, these horizontal lines. And there was, I'm showing you two plots here, percent saturation as well as concentration. And so there was an exceedance of this contingency plan threshold in percent saturation only, not concentration. And at this one station in the Stellwagen Basin. And so this is a station that's not in the near field near the outfall. And we don't believe that this result is connected to the outfall. If it were, you might expect low conditions, low oxygen near the outfall, and we didn't see that. It's also an exceedance in percent saturation only and not concentration. And concentration is, a lot of people consider concentration to be more important from a biological standpoint than the percent saturation. So again, there's no hypoxia in MassBay or Stellwagen Basin. The concentrations are up above 6 milligrams per liter. The state water quality standard is down at 5 milligrams per liter. We're not near that. However, there is definitely an indication that during this year, oxygen concentrations are lower than they have been in the past. And I think this is part of a long-term trend. We've seen this in the rest of our data set region-wide, and it's similar to some of the things that Amy just described. So this is my summary slide. How am I doing? Do I have time to go through it, Prasada?
Yeah, about 30 seconds.
Okay, well, why don't I just skip this? I think it's fine. We'll move on. And great. I'll be happy to answer questions in the chat.
No, absolutely. Thank you, Dan. And I feel bad because we don't want to shortchange any of the presentations by any means. And I feel like we could dedicate like a whole hour to each of these presentations and to really dig deep into them. But thank you. That was really helpful. And people who have chance to ask questions during the panel discussion, etc., so we can get into these a little more. But there are some questions for you, Dan, if you want to look at them in the Q&A. Next up will be, we have Dr. Jeffrey Barbaro from USGS. Dr. Barbaro has been working on, he's a hydrologist who's been working on impact of groundwater, especially on the Cape. And you can take it from there, Jeffrey.
Okay, can you see my screen?
Not yet. You have to click share screen at the bottom.
Okay.
Do you see the green button? Yes. Yeah, perfect.
How's that?
All set.
Okay, I will be fairly quick as well. I'm gonna address the issue of groundwater discharge and nitrogen discharge, and we don't have any studies focused on Cape Cod Bay. We just have not had the opportunity to really do any of that work. So I'm going to focus mainly on some of the tools and methods that we have available to us. First, I'm going to talk about groundwater discharge itself, and then I'll say a few words about nitrogen. But first, one slide on groundwater hydrology of Cape Cod. I'm probably just people here in the meeting who don't think about groundwater very much. So the panel on the right here, the plan view map. This is a map that shows up in quite a few USGS reports. And what it shows is that the Cape is actually composed of 6 different discrete groundwater lenses. They're separated by surface water divides. They kind of function as independent aquifers. They all have mounds in the center, and groundwater flows radially off those mounds toward the coast. They're called, I don't know who named these, but they're called Sagamore, Monomoy, Nauset, Jaquesset, Pamet, and Pilgrim. And in cross-sectional view, the lower panel here shows the situation that occurs mainly on the western part of the Cape, probably right around through here. You have precipitation that provides all the fresh water, ends up as recharge, and groundwater flows from high water tables to low water tables. So tends to flow from the center of the Cape outward to the coast, to rivers as well. And fresh water it goes all the way down to bedrock. So it sits on bedrock. As you go further out, the fresh water does not go all the way down to bedrock, and you've got salt water underneath fresh water everywhere. but you have the same situation where groundwater flows from highs to the coast. So how do we get at this question here of groundwater discharging to the coast? How quantifying that rate? Well, I would argue that one of the best ways to do that is with the groundwater flow model. And we have a number of regional groundwater flow models that we've developed over the years, USGS. On the left side here, left graph, mainly just want to call your attention to the boxes here. These are model domains for groundwater models we've published. So we've got a model of the Plymouth Carver, western part of the Cape, Central Cape, Outer Cape. There's been various generations of these models done over the years. In particular, this one here, we've got multiple generations of this model. And the most recent one was published in 2019, which is the most sort of state-of-the-art model we've got for this part of the Cape. And then on the right, this shows some of the information you can get from these models. So this might be a little bit hard to digest, but what this shows is that a parcel of water that recharges the aquifer right here in this orange area is going to discharge directly to the coast. Water that recharges in the green area is going to first discharge to a stream, and then it's going to, of course, the stream is going to flow out to the coast. And the blue areas are groundwater that's first going to flow into a kettle pond and then either back out into the aquifer or into a stream. So these areas here, these are the areas where groundwater is going to flow directly to the coast and discharge to the coast. One of the other really, and this is the main point I want to make about these models, is that one of the things that comes out of a calibrated groundwater model are hydrologic budgets. So this is from a summary that John Masterson produced a few years back. And these are the aquifers here. So this is the Plymouth Carver. These are the 6 Cape Cod lenses. This is inflow. So this is long-term average, like a steady state inflow of water into these aquifer systems. And then what he did here is he broke out the discharge to the major receptors, call them ecological receptors, surface waters, and he's broke these out into percentages. So for example, if you look at the Sagamore here, 270 million gallons per day in on average, 66% of that water discharges directly to coastal waters, 28% to streams. He's lumping streams and ponds here, I believe, and then about 6% to wells, mainly the large water supply wells. These, a lot of this water goes right back into the aquifer as treated or untreated wastewater. So then you can see here differences. As I mentioned, these function pretty independently. So you can look at, for example, the Chequesset, which is an outlier. About 50% of the groundwater discharges to streams versus smaller percentages here. And the reason here is that the Herring River basically bisects the Chequesset lens right across the middle of it. So these are totals for the entire aquifer system, but these rates, these could be expressed as rates. These can be distributed to all the model cells along the coastal boundary of the model, so you can get more localized information about groundwater outflow rates to the coast. The other thing I'd mention is these are for large scale regional models, but you could construct models at the basin scale, at the sub-basin scale, and at the local scale, and get similar quantitative information about freshwater inflows. Well, freshwater outflows of the groundwater flow system, inflows into the estuary or coastal waters. I'm not going to say too much about this. I just wanted to mention that there are definitely a lot of other methods that are used to understand groundwater discharge. I will say that once you start to get to the embayment scale and the site scale, things get complicated really quickly. It's at 10,000 feet when you're using a groundwater model. It's, you know, you can calculate how much groundwater is coming out. When you really start to look at the details, there's a tremendous amount of complexity. I've grouped these by sort of major techniques that are used. Geophysical methods, these methods are good for not only, well, mainly for determining the subsurface geology, and which can exert a really large control on where the fresh water is coming out. You can also image, where the fresh water is. I'm using Cape Cod examples here. This is the Yale River in Falmouth. There's many thermal methods, a huge amount of work that's done. This happens to be an example for a Shumat Pond in Mashpee. And then there's also sort of more conventional techniques where you get out there in the water and pound piezometers in the ground and try to take water quality measurements and other hydrologic measurements. There's been a lot of really good work done in Cape Bay, sort of offshore work. And I would say generally that these methods are better at determining the spatial distribution of fresh water rather than the actual rate of discharge across the interface. But these are often used in combination with each other. So for example, you might use thermal methods to really map out where the groundwater is coming out, then go into those areas and use other techniques to try to determine rates of groundwater outflows. Now, the final point here is that this cartoon here shows in situations where you've got tidal activity, these little cells set up from tidal pumping. This is one conceptual model. And this becomes a really complex interface. It's really dynamic. It's really tough to work in my opinion. And I'm sure maybe other researchers who work in this environment may disagree with that. But this is a complex interface. And if you're really trying to measure the actual flux of groundwater as well as nitrogen, it's a difficult thing to do. So I'm just a couple slides on nitrogen transport and discharge, and I'm just going to use one modeling example from another USGS study by Don Walter, and it's in the Popponesset Bay watershed, which is over here in Falmouth. And again, you get a lot of high resolution information about where recharging groundwater is discharging. So this is the watershed boundary here, and he actually constructed a sub-regional model from the regional model to develop a more high-resolution model. And this blue area shows groundwater recharging the aquifer here, discharges into the ponds up here in the headwaters. The green area, groundwater discharges first into the streams, which then of course flow down into the bay. And then down here in the lower part of the watershed, all the groundwater recharging in here discharges directly via groundwater pathway to Popponesset Bay. This is kind of a complicated slide here. The point I want to make here is just to sort of show how a study like this would be done. So what it does is it combines information on nitrogen sources on the landscape, inputs, with a groundwater flow model to develop nitrogen loading curves at ecological receptors that the user defines. So the purple here are nitrogen sources, and this is down to the parcel scale. So these are individual homes that have septic systems. Calculate the loading for individual septic systems. I believe there's fertilizers in here as well. So you have this really a pretty good understanding of where the nitrogen sources are within the watershed. And this is sort of your source function for your model. And then what the groundwater flow model provides is a distribution of travel times, because of course groundwater moves slowly. There's a lag there that sort of gives rise to the legacy nitrate issue in groundwater. There's a reservoir of nitrogen in there. And what this shows is to these various defined endpoints or receptors, it shows the distribution of travel times. And what this happens to show is that most of the travel times are short. So surprisingly short. So the median travel time, about 4 years from the point of recharge to streams, and then about 6 years to estuaries. There are some stragglers. There's some much longer flow paths where groundwater flow path is quite deep and long to get to the receptor. But this travel time information, along with the source information, then produces loading curves at the endpoints. And this is a complex study. There's a lot of nuance here, but I just selected a couple just to make the point that, for example, if you look at the loading, and this is in kilograms of nitrogen per year, it starts to approach a steady state. And in this, what he did here is he actually ran a scenario where the entire watershed was sewered. So all the sources, all the septic system sources were shut off, and you can get an understanding of the rate at which the nitrogen starts to fall off. The nitrogen load starts to decline. So the power of this approach is that you could run scenarios, look at do management, various management actions, and then see the results. The final point I would make here is the issue of nitrogen attenuation. And what he's doing here is this is actually the load to the Mashpee River itself. And the blue curve is completely unattenuated, no nitrogen losses at all, conservative transport of nitrate. And then these two curves here are attenuated loading curves, where I believe you use the MEP attenuation factors in the ponds and in the rivers. And the main point being that there's quite a large difference there. And determining how much nitrogen attenuation there actually is within the aquifer itself, within, in the watershed, as well as that complex mixing zone right at the end of the flow path is one of the more challenging aspects of really determining the actual nitrogen flux to the coast. So that's all I've got. These are the main points I was trying to bring out. We do have groundwater models for the entire coastline bordering Cape Cod Bay. There are various vintages, and it would take some work to actually piece them all together and to do an analysis, but it's possible. Also, these models, they do provide estimates of groundwater discharge to coastal waters. And then, again, using modeling combined with nitrogen source data is another technique that's pretty powerful for trying to estimate nitrogen discharge to the coast. And that's all I've got.
Thank you very much, Jeffrey. It was very insightful. It's good to know there are so much data about groundwater, which is such an important issue on Cape Cod. So the last speaker for this section is Dr. David Workman. And after his presentation, we'll break up for a very short break. We're trying to make up time. So David Workman, comes from the Department of Environmental Management in Rhode Island, and his research is specific to harmful algal blooms and phytoplankton. And today he's probably going to touch on the issue of Karenia and underage trees, Cape Cod Bay. Take it from here, David.
Let's see, can you see anything yet?
No, yes.
How about now? Okay,
yeah.
Bear with me for a sec.
That's fine.
How about now?
Yes. Yeah.
Okay,
perfect.
Great.
Yeah.
Great. Well, thanks very much for this chance to talk to you about Karenia mikimotoi in Massachusetts Bay. I have about 10 slides, should be able to get through them pretty quickly in 10 minutes. And my goal here is to fill in a few details on the ecology and physiology of this organism, its history in other parts of the world, and some recent Massachusetts Bay observations of this organism. Okay, so my involvement with the project is through MWA monitoring. I've been doing the phytoplankton species IDs and count since 1992. And just briefly, we cover now 10 stations in Massachusetts Bay. You can see some of them on the map. Amy and her crew down at PCCS collect and I count up phytoplankton from 3 additional stations along the Cape. And it's about once a month, February to October. And we've been doing this actually a little more early on, but this type of coverage since 1992. So that's the background. That's the data source I'm going to be talking about. Okay, so Karenia mickeymotoi, it's a dinoflagellate. There's a photo micrograph of it up in the corner here. It's a medium-sized phytoplankton cell, about 30 micrometers in diameter. It's a dinoflagellate, and it's athecate. That is, it does not have a series of thick cellulose plates around it. It's unfortunate in a way because those thick cellulose plates are a guideline to a scheme to identify species of dinoflagellate. So there's been taxonomic confusion, especially in our region of the world, as to what exactly this species is. For many years, since the 1950s, a similar species called Gyrodinium aureolum has been identified in this area. So there's a history of back and forth identification. I can tell you that all the cells observed in Massachusetts Bay since 2017, those observed in Maine since 2017, are morphologically consistent with Karenia mickeymotoi, and there's been at least one molecular ID as Karenia mickeymotoi. So we're pretty confident on the recent ID as Karenia mickeymotoi. It historically bloomed in Japan. That's where it was first identified, then the North Sea, other parts of the world, including up and down the East Coast, recently in Alaska. It's eurythermal. It has a wide temperature tolerance. Harmful effects. It doesn't have any direct human harmful effects. It's not a biotoxin producer that we know of that's going to harm people if they eat shellfish, for example. There is evidence of toxin production. Gymnosin, that's ichthyotoxic, it kills fish when it's in high concentration near fish. It's also allelopathic. I'll talk a bit about that after. Its big effect is it's a biomass bloomer. This image down at the bottom is from Save the Harbor. That's Boston Harbor in September 2019. It looks like a cup of coffee. So when it blooms in these high numbers, it's very visible. And as you've heard from all the speakers so far today, earlier today, it's a biomass bloomer. When that biomass decays, bacterial respiration draws down dissolved oxygen. Okay, next slide, hopefully. So just repeating, the main harmful effects are through these high biomass blooms crashing, causing anoxia, especially for benthic infauna, right? Things that can't swim away, they're stuck in the bottom. They're going to die from lack of oxygen. Around the North Sea, the coast of Scotland and Ireland, oh, sorry, page up. Caged finfish, right? They can't get away. If a bloom of Karenia gets into a cove where there's a caged fish operation, lots of times there's mortality associated with anoxia. This headline over on the right side, that's from 2017, the Clam Flats, parts of Casco Bay, Maine, lost about $250,000 worth of shellfish in that event. And there's similar examples around the world. Okay. Malcolm touched on this stuff. I'll just go over it quickly. The thing to keep in mind is that this organism is well suited to life in the subsurface layers, the pycnocline. Low light, sources of nutrient, it can migrate up and down to get to them. It has a high swim speed, which helps it form patches that can be transported horizontally thousands of kilometers in thin layers. I said this already, it has a low light requirement. It's mainly autotrophic. It has chloroplasts, right? So it needs light, but it also can switch hit a little bit and consume bacteria as a nutrition source. So it's flexible that way. It takes up all forms of inorganic nitrogen, but has a preference for ammonia. It's just energetically more efficient. For example, it can take up ammonia both day and night. It can only take up nitrate during the day when there's energy from the sun. Lots of times people associate red tides with explosive growth. It's a moderate growth rate, 0.5 to 1 division per day. So think of it like a bank account. If you're not able to build your bank account by having a great income. The other way you can do it is by saving money, having low loss rate, right? Don't spend as much. That seems to be Karenia's strategy. It's allelopathic, so it kills off other cells. Grazers don't like it, so it's not eaten much. And has a mechanism that was recently discovered in 2020 to overwinter as a dormant cell in the water column. So it's a small cell, but it's very, very carbon and chlorophyll dense compared to a diatom that has lots of cell sap and vacuoles to have some density control. This cell is packed with chloroplasts and carbon. So they're small, they can get to high numbers, and there's a lot of carbon in each cell. On the right, this little graphic shows the effect of the presence of Karenia, some chemical compound that it gives off infiltrate from Karenia. So the white dots, it's the Thalasseosaurus pseudonana growing a normal exponential growth curve. The black dots or squares is growth rate when exposed to Karenia. So there's something about it that when it reaches high enough densities, right, that's 10 to the 5th per mil, that's much higher than we see in Massachusetts Bay, can impede the growth of other cells. So its strategy is not so much explosive growth, but avoiding being eaten and outcompeting other phytoplankton, it seems, at low light levels in the pycnocline area, subsurface. Okay, so historically, this old paper down below, there's been a shade floor, that is a subsurface group of phytoplankton species that do well at low light. It was there when Bigelow and Lillick and Fish were looking at Massachusetts and Gulf of Maine phytoplankton 100 years ago. We see it historically in Mass Bay. Historically, that layer is dominated by another dinoflagellate, Serratium. And here's its abundance every year on average in that series of stations off Boston from 1992 to 2020. And it's cyclical, goes up and down. And historically, up until 2007, there was a good correlation between how stratified the water column was in the early summer season and the abundance of Serratium later. That seemed to change in 2007. It kind of falls apart after this. So maybe something similar to what Malcolm showed about the changes in sort of the physics of the system. And it seems like in the past 5 years, Karenia mikimotoi, is a new member of this subsurface chlorophyll max flora out in Cape Cod Bay and Mass Bay. Okay, a little bit more background. The species was first identified in Japan in the 1930s. It's well adapted for life and movement along coastal fronts, and it has a history of unexpected appearances all around the world. It was mainly known from Asia in 1966 off Oslo Fjord in Norway, a place where people had been counting phytoplankton and studying them at that time for almost 100 years. It appeared unexpectedly, and it's repeatedly shown up in places unexpectedly. Most recently, the coast of Kamchatka, about a year ago now, where it killed benthic organisms around a 1,000-mile stretch of coast there. So it has this history of popping up in various places where it hasn't been seen before. Okay, from Massachusetts Bay in our part of the world, as I said earlier, it's been identified in the area of Woods Hole and the mouth of the Narrow River since the 50s as G. aureolum. There were regional appearances of cells that were identified as G. aureolum that look an awful lot like Karenia mikimotoi from the 60s, from North Carolina up to Maine, including blooms off New Jersey that killed benthic organisms. A bloom of similarly Karenia mikimotoi-like cells in the Gulf of St. Lawrence in 1993, in Maine in 1988. But there's this sort of uncertain background of its presence in the region. But it is very clear to say that from 1992 to 2016, in regular monitoring in the Massachusetts Bay region, it was not observed. In the summer of 2017, it appeared synchronously along 100 miles of coast from Portland, Maine, this headline here, down through Massachusetts Bay, the MWA monitoring area. So it showed up all at once. That's my timer, so I'll finish up. And it's hung around since. So let me just skip. Here's the first few years of when it showed up. You can keep track of how many days of the year it was observed in the monitoring. Two things, the red dots are the subsurface chlorophyll. It's much more abundant. 4 to 30 times more abundant at the deep layer than the surface. Numbers approaching 1 million per liter, 2019, 2020. This year's sort of a low year so far. Again, wrapping up the same stuff. By 2020, the cells are around pretty much from February to October. And so far this year, I've only finished through the end of August. It's the same story. So wrapping up. Corenia micomotoi has been in this area since 2017. It's shown up each year since then. It's in the pycnocline. It seems like around 1 million cells per liter is where harmful effects start happening. Along the lines that Malcolm outlined in Ireland, 1 million cells per liter is equivalent to about a 1 milligram per liter oxygen demand. So for the future, if it hangs around at these levels, you can count on perhaps that level of added oxygen demand on top of the climate change, the warming that Malcolm described. It may be pushing things over the edge. So thanks very much. I'm happy to answer questions by chat if people have them.
Thank you, David. It was interesting to know about this less known species until more recently. Pam, shall we proceed to break?
Yes, I think what we'll do is have a break until 4:10. And I'm going to open up the chat again. If you didn't get a chance to put your name and affiliation in the chat, continue to ask questions. I think what we might do to make sure that everybody has a chance to have some input is that we may try and kind of mash together the panelist discussion and the participant questions. That will make sure everybody has a chance. So don't go way too far, and we'll see you at 4:10. Thank you. So we're getting ready to get started again. If you've stepped away, we can have you scurry back to your seats.
Hi, Pam.
Uh-huh.
There? Okay.
Yep.
Sorry. Were you the one talking?
It was, yes. Just
Sorry,
calling people back.
I was coming back.
Okay, good. Just getting everybody back in line. So our final formal presentation is from Mark Tedesco. He's the director for the Long Island Sound National Estuary Program, our counterpart down there in Region 2 EPA in Connecticut, New York. He's going to start us thinking about next steps based on their experience in Long Island Sound. So go for it, Mark. Thank you.
Thank you, Pam, and good afternoon, everyone. So I've been asked to give a case study in responding to hypoxia in Long Island Sound. My subtitle is The Good, the Bad, and What's Next? And I'm going to obviously skim the surface of a lot of work and ask for your patience on that. And let's see if I can just go to the next slide. Okay, so where we started, hypoxia was widespread in Long Island Sound. This is a map from 1994. The area in red and black are concentrations below 2 milligrams per liter, critical levels as identified earlier. After a lot of work, modeling, monitoring, EPA working with state of New York and Connecticut developed a total maximum daily load to reduce nitrogen loadings from New York and Connecticut by about 60% with some additional reductions from other sources. So how are we doing? This graph shows the point sources from New York and Connecticut, the predominant load, nitrogen load to Long Island Sound. The decreases total to about 47 million pounds per year less in 2020 compared to the baseline discharges. And the discharges are now meeting the waste load allocation that was identified in the TMDL. That represents about $2.5 billion worth of infrastructure investments. It's not just point sources. This graph represents work from USGS in monitoring the major tributaries draining to Long Island Sound. Obviously, in the blue line, you see the annual variation from year to year, wet to drier years. The red line shows the flow normalized load. So we can see that overall decreasing trend, again, attributed partly to point source upgrades, but also to non-point source reductions throughout the watershed. Part of that non-point source reduction is from decreased atmospheric deposition experienced broadly throughout New England. This represents the precipitation weighted concentrations for NADP sites in the Long Island Sound watershed. And we can see the levels of the decline in concentrations from the 70s up to more recent periods. So big reductions in atmospheric deposition contributing to those reduced watershed loads. So in summary, big reductions meeting or exceeding the TMDL in the wastewater treatment plants in Connecticut and New York, big reductions in atmospheric deposition. We infer similar reductions in agriculture based on surveys of numbers of livestock or fertilizer applications through the watershed. And what is remaining are the more difficult sources to control. Urban stormwater, we've had an increase in impervious area during this period of time, increase in septic waste from wastewater not connected to centralized treatment systems. Has been an increase in the basin population, and that increase has been greatest in non-sanitary sewer connected areas of the watershed. And then also, again, an increase in turf and grass. So The question now is, well, from those reductions, what are we seeing in terms of dissolved oxygen trends? So this graphic shows the maximum area of hypoxia. This is less than 3 milligrams per liter, 1987 to 21. Obviously, year-to-year variations in large part based on some of the weather conditions that speakers had highlighted operate in Cape Cod and elsewhere. But importantly, if we look at that 5-year running average, the average hypoxia, that average size of hypoxic areas in Long Island Sound has decreased in half from the average from around 2000, the pre-TMDL average. So we are seeing that positive response in dissolved oxygen to those nitrogen reductions. Here's a question mark, something that would we need to consider in terms of future steps. And that's while nitrogen mitigation has reduced hypoxia, the improvements that we would have observed have been dampened by warmer water temperatures. And again, as speakers emphasized earlier, what's true for Cape Cod is true for Long Island Sound as well. We have a trend of increasing water temperatures about 2 degrees Fahrenheit over the past 20 years. And as a result, we estimate that the improvement in dissolved oxygen has been dampened by about 25%. Further, if we forecast out predicted future increases in water temperatures, we could see an erosion of about 35% in the current levels of dissolved oxygen. So we are not yet achieving water quality standards. In part, that's been, the progress has been held back because of increased water temperatures, but it could be further eroded in the future. The other aspect that we're dealing with now is that in spite of the progress, there are still impairments. And it's not just the open water hypoxia, harmful algal growth that could impair recreational uses, reduce light for critical habitats such as seagrass, and then nearshore diurnal hypoxia events that may also impair the system and result in fish kills. So while we're decreasing the open water hypoxic area and severity, hypoxia still remains in the open waters of Long Island Sound, and there are other eutrophication impairments that we're concerned with. And that has led us now to our next step. This is the what's next part of the presentation, and leading to some fundamental questions. Are further nitrogen load reductions needed to meet water quality standards? What about the impacts to nearshore waters and embayments? And so we've embarked on a major program to expand the monitoring and modeling. A new ROMS RCA coupled system-wide model is being developed. We've done analysis on existing data to link nitrogen concentrations to impairments in Long Island Sound. We're viewing more difficult to control and less previously well-defined sources, such as on-site wastewater systems. We're improving the overall nutrient load estimates, working closely with USGS and New York and Connecticut to improve the tools for the nitrogen load estimates. And again, continuing the program of research, monitoring, and modeling. I won't go into this other than to highlight the extensive monitoring program that is underway in Long Island Sound, monthly, year-round monitoring, as well as bi-weekly during the summer. Both open water embayments and tributary. And this is really, again, the summary of what's next. We're going to look at further nitrogen load reductions. We've, again, expanded efforts to model the flow of nutrients, associated impacts, address deficiencies in existing regional model tools, support state and local efforts, as well as the regional tool development, improve data sharing, visualization tools, with the goal, ultimately, again, of evaluating the 2000 soundwide TMDL and to set sub-watershed nitrogen targets that are protective of local water quality and address those local impairments. I just put this slide in to show some of the work that we're collaborating with USGS on. Jeff Barbaro highlighted some related work in Cape Cod, similar to work underway now in the Long Island Sound region. I am not going to detail this work at all, other than to highlight that these partnerships are proceeding. And with that, I was going to stop and try to give us as much time as possible to have the panel discussion. Thank you.
Fantastic. Thank you so much, Mark. This is really helpful as we look to try and figure out what's happening in our system and where we might find some lessons learned from others. And I wanted to get our conversation started off by just asking all of the speakers and panelists to turn on their cameras so we can see everybody. And then we'll quickly have the people who are joining us now just introduce themselves that you haven't heard from yet. So Kathy, can you say who you are?
Yeah, so hi, I'm Kathy Vakalopoulos. I am from Massachusetts Department of Environmental Protection. I work in surface water discharge permitting. So we work with EPA to permit point source discharges, including wastewater treatment plants and industrial facilities.
Fantastic. Thanks, Kathy. Leldon?
Hi, good afternoon. I'm Leldon Langley with Mass Department of Environmental Protection. I'm Director of Division of Watershed Management. So my responsibilities include the wastewater programs, meaning septic system, groundwater discharges, and the surface water discharge programs, water management program, which is a large-scale water withdrawal program, and the watershed planning program, which is our monitoring and assessment program that also develops TMDLs.
Is that all, Weldon? I can't.
For now.
Joyce?
Hi, I'm Joyce Novak. I'm the Executive Director of the Peconic Estuary Partnership. That's the National Estuary Program on the east end of Long Island, sandwiched between the Atlantic Ocean and the Long Island Sound. And I'm here to offer insight from another place where groundwater fed systems. So we have some of those similar issues.
Thanks, Joyce. Ken.
Hi, I'm Ken Moraff. I'm the Director of the Water Program at EPA's New England office, and the program includes NPDES permitting. So one of the things we need to do is translate complex science like what we've just heard about into sound regulatory decisions. So I really appreciate all the effort that went into today's discussion and looking forward to hearing from the panel.
Thanks, Ken. We did have Beth Cassoni from the Massachusetts Lobstermen's Association was supposed to be here, but she has no power and no way to get to her work because of downed trees and everything. So we'll definitely follow up with Beth after the forum.
Sorry
yes.
to interrupt. Rocky Geyer, same thing. He lost power and his internet and had to leave.
Okay,
Sorry,
thank you. I thought we were missing somebody. Thanks, Cressida. So because we did the awesome presentations went on a little bit longer than we thought, and I have no regrets because there's so much good information. I think what I want to do is think about combining a little bit the discussion from or questions from participants along with the discussion from the panel. So I'm going to do my best to facilitate that. But I do want to start out with kind of a reality check from the speakers, because it does sound, and Jeffrey Rosen pointed out in the questions, it does sound like there's at least less documentation of nutrient issues directly causing hypoxia than it is the Karenia coming in and taking advantage of what nutrients just happen to be there. Does anybody want to comment on that? I know Malcolm, you spent some time on that, and Jeff. I mean, David.
Yeah, I'm happy to comment on it. I'm going to speak specifically to Southern Cape Cod Bay, where we've really spent most of our time looking, and it really does seem like the anomalously high chlorophyll fluorescence is the cause of the hypoxia. And we really think strongly that that probably is Karenia, given everything that we know. The nutrient data, if anything, the key result we see in the nutrient data is the bottom nutrients have been utilized. And we think that's really what's different, is that those nutrients are there. And they just prior to 2019, there was not, the physical conditions weren't, the physical conditions weren't allowing the organisms to use them. And that, we think that changes in the physical environment is why Karenia is doing well. You know, it likes deep stratified environments. And so we think that's basically why it bloomed in the last 2 years, or 2019, 2020.
Thanks, Malcolm. Does anybody want to counter that? Anybody on the panel want to raise a counter argument to that? I see Rocky's back. Thanks for joining us, Rocky.
So I don't want to counter that. I want to support it, because I think, and Dan can say so too, with the ambient monitoring that we do at the center and at MWA, we aren't seeing any increases in nutrient concentrations, either in surface waters or along the shoreline consistently or at depth. So I wouldn't point to nutrients as one of the factors.
Mm-hmm. Mark and Joyce, have you looked at all at phytoplankton as a factor in your hypoxia events?
Well, certainly, I mean, phytoplankton production, which is higher in the Western Sound, exactly where we see the worst hypoxia, and that's the link. It's nutrients to phytoplankton production. We have not seen the kind of major shift in, for example, highlighted in Cape Cod with Karenia. So we have not had bloom organisms take over. There have been some shifts in phytoplankton, but it hasn't been sort of a HAB-dominated system. We've had some localized Alexandria blooms, but it's more straightforward, or appears to be straightforward, nutrients to phytoplankton production, organic matter production.
Mm-hmm. Joyce, did you want to add to that?
Not really. We have a HAB problem for sure, but we don't have a Karenia issue at the moment until it potentially comes south. But yeah, for now, that's not an issue for us.
Hasn't been identified as the issue. Okay. So let's I'm going to go out on what's seeming like a more solid limb than I thought it might be earlier. So let's say that it is a Karenia problem. Is there something that we can do? Do we need to do more research? Do we need to collect more data? Do we need to figure out how to disrupt this cycle that we're in? What are some responses that we could take to the situation that we're seeing?
Well, I'll jump in again. You should always do more research, and that's obviously a biased perspective, but more research is always good. But I think in the case of what's going on in Cape Cod Bay, I think the program that Tracy has organized, working with the local watermen, is really a cutting edge example of how you integrate the people utilizing the resource. You help them figure out where not to put their pots to try to minimize damage to the resource. And I think I don't think we're going to disrupt Karenia. There's nothing we can do to prevent it from blooming, but we can work with fishermen and people utilizing the resource to minimize the damage to the resource. And I think that's really a great example of how what Tracy's doing with the pot loggers and developing that resource for the lobstermen, I think is a great example of what can be done.
Mhm.
Yeah, I'll follow up on that. I think that the willingness of the fleet to get involved and their desire to know what's going on, the more we can give them back the information, I think that will let the users, both the study fleet participants and the rest of the rest of the lobster fleet in the area, start to understand what's going on and start to sort of say, we're not at the level now where I need to think about moving my gear somewhere else. Because it doesn't help them to pull up traps full of dead lobsters. It's a waste of money and time on their part, and obviously doesn't help the resource. So I think they have a pretty big vested interest in paying attention to this and to participating and moving forward. And that's certainly the feedback I've gotten from the fleet.
Thanks, Rocky.
Yeah, just a couple things to add to that. The one thing that I think is great about the situation we're in is the way the MWRA program has provided us with this context that we know what this is not, and that it's not. This is not something that people were really afraid of at the beginning of the outfall pipe, that this big green blob was going to come down from Boston and kill everything in Cape Cod Bay. And because we can see that this isn't simply an ammonia spike from the outfall that's inoculated Cape Cod Bay. And and there's many other things that we know it's not. And we don't know exactly whether it's Corenia. That's a pretty good hypothesis. But I think another thing that sort of backs up what Tracy and Malcolm said is that if we know that it's going on and we know where it's going on, then the fleet can respond and we can really keep there from being significant harm from this so far, unless it changes, unless the breadth of it changes. But right now it's a pretty localized, when it happens, it's pretty localized. And so that's something that the fleet can really respond well to. The way that lobsters die is when the traps are where the hypoxia is. And so if they pull the traps where the hypoxia, they just don't put traps in where the hypoxia is, then the lobsters can swim away.
Yeah, and I'll ask the panelists and speakers to keep trying to answer questions that come up in the Q&A too, so that we can try and maximize our time. Thank you very much for that. That's really helpful. And there was a comment earlier that, well, and Barbara Moran points out too in the question that, is it just a factor of things getting warmer, so Corenia has opportunity or it's easier for it to be here. I know that they have the wide range in which they're active, but there is that sweet spot where they form blooms. Are we kind of fighting against climate change here? And then Carl Person earlier had asked if getting in there and breaking up the picnic line might be an option, which sounds like a big issue. Or, I mean, he was talking about reoxygenating the area, but I would think that you could also think about doing some more artificial north winds that were pointed out earlier. You can tell that I'm just like the dreamer kind. I'm not basing this on anything that I've studied. So push back.
So Pam, if I can jump in just 2 seconds, I think I agree from sort of an outside perspective with the arrangement with the lobster fishermen. I did work on the western coast of Ireland with the Azimuth Project, which is a HABS warning system for the western seaboard of Europe. And it was geared towards salmon fishermen in cages, salmon cages. And they found with Corenia, that's what was one of the reasons, and there was a Pseudonychia diatom that had driven this, what can we do? Waters are warming, things are migrating. At some point, recognizing what you don't have control over, and developing warning systems to do that, where to put cages in, where to put, drop your pots, do that type of thing to minimize the damage. And I'm always weary of that notion that we can, you know, heavily engineer a system to fight against it. So I'll just throw that out there.
I hear you, Joyce. Anyone else? Ken?
I don't have an answer to that, but I do have a question if we're ready to move to a new topic. I guess
Please go.
people who are, the experts who are more familiar with how this has played out in other parts of the world where Karenia has been around longer, does it, I guess, two things. One is, right now it seems fairly localized here, does it tend to spread? Does it start this way and then it's going to move all along the coastline? Or not, or is that not clear? And second, just still thinking more about the nutrient issue, and I understand the evidence against the idea that nutrients cause this, but elsewhere in the world, do we see a link between high nutrient concentrations in Karenia? Does it tend to happen more where there are higher, where there's elevated nitrogen or not?
Who wants to take that one on?
This is David Borkman. I can't get my camera to work for some reason.
Oh.
But to address your question, I think a good example is the North Sea example. Let's see if this works. So the phytoplankton of the North Sea was well studied from the late 1800s through 1966 without seeing this organism. And then it showed up out of the blue in 1966. Since that time, where it was first seen in Norway, Oslo Fjord, it's now a regular component. And earlier, people spoke about the need for a HAB early warning program. So now all around Scotland, Ireland, and England, it's one of the, just like we monitor Alexandrium here, they monitor Karenia. So when it's showing up in a front along the cove where your salmon pens are, or your aquaculture facility is, you take action, move the pens, harvest what you can, things like that. So their example is relevant because they're similar water temperatures, fronts with advection of Karenia blooms into shallow areas, like the example Malcolm and Rocky have outlined in Cape Cod Bay. I mean, it's when it's out in the front and it's several out along a coastal front, it's a few hundred meters deep, it's not that big a deal. It's when these layers of cells get blown in, injected into places that are less than 50 meters deep, and the cells end up near the bottom and suck up all the oxygen. So, and on nutrients, I think the legal term necessary but not sufficient applies. Obviously, they need nutrient. They have to get it. They're very good at taking up ammonia, so that means they can live on a microbial loop, right? Any little dribs and drabs of ammonia that's being recycled, they're super good at taking up. Anyway, just some thoughts to get at some of those questions.
Thank you, David. So I'm trying to go back to some of the other questions, or anybody has their hand raised. There were some, there's some back and forth with Ken K. Would Amy or Malcolm, would you like to talk about this question from Ken?
Yeah, I'm happy to just provide a few thoughts. The comment really is about linking the high chlorophyll fluorescence to Karenia, and I agree that that's we don't have as much data as we need to do that. However, I will point out that, and I'll also highlight that getting some of these layers are on the order of 1 meter thin, and so getting a water sample from that layer is a pretty challenging logistical observation to make. But we did do that this year, and we did one sample last year where we got water from what we think was close to the chlorophyll maximum. And then the one we got in 2020, that was the sample that had 700,000 cells of Karenia per liter and was more than 70% Karenia. We got one more this year, and David Borkman can correct me if I'm wrong, but we got one from a chlorophyll max this year, and it had over 1 million cells per liter when David did the count. So we do have some actual data, and then we have a lot of circumstantial data suggesting that the high chlorophyll that we've seen is Karenia.
Yeah, just to follow up on that, Malcolm, the sample that Amy and all of you were able to find in that hypoxic zone, which was much less widespread this summer, 21, was 1.3 million Karenia per liter, and it was dominant in the phytoplankton. So yeah, we don't have a lot of individual hypoxic layer Karenia counts. We have the monitoring showing it show up. We have the counts that show up much higher at the subsurface max, and then the handful of the recent work you guys have done down in Cape Cod Bay.
So let me ask a question then, because Tracy, you said you didn't observe as low DO concentrations this year as you in 2021. Did we look at the chlorophyll at the same time? And is there any indication that this is like a glitch and it's gonna bounce back to normal, or how is that looking?
So this was a question more for, I think, Amy and Rocky probably, but with the study fleet sensors, those sensors only log dissolved oxygen and temperature. And the Center for Coastal Studies surveys that they do, we sort of use the study fleet data to trigger what we're calling these higher resolution surveys that CCS goes out and then does on a much more frequent basis than they normally would do their monitoring. And it's on those surveys that they're able to get the profiles that include not only the DO and the temperature, but the chlorophyll max data. So I'm going to punt this one.
Good, thank you. Sorry about that. Amy,
No
now
worries.
we're going to put you on the spot instead.
So at the beginning of 2021, things were setting up pretty well that we thought we were going to get another event. I think actually dissolved oxygen lowers at bottom at depth were lower at our stations than we had seen in 2019 or 2020 or maybe ever before. And Dan again can speak to the MWRA data because they saw the same things at the stations that we sampled for MWRA, FO1 and FO2. So we were thinking it wouldn't happen, but again in 2021 we didn't see the high chlorophyll levels that we saw in 2019 and 2020.
And I'll just jump in and add one more thing. And David alluded to this. We did start to see hints of really, really high chlorophyll, and we were able to get samples from it, and it was Karenia, but that just never turned into a widespread bloom that persisted like we saw in 2019 and 2020. And and trying to predict blooms of a HAB is extremely difficult to do. So we don't know exactly why it didn't happen, but we suspect that the fact that the oxygen never bottomed out has to do with the fact that the bloom never developed.
So Tracy, and then Kathy has her hand raised.
Oh, I forgot about the hand function, sorry.
No worries.
Yeah, so we were, like Amy said, that we were seeing those lower than expected values in like mid-August this year, and we totally thought that was going to continue to decline. And those low levels in the middle of August are what DMF issued the advisory on that some of you probably saw, with the expectation that, oh boy, this is starting earlier than we've seen it start. This is going to get bad. And then, like Malcolm and Amy said, it just didn't develop for whatever reason. So we were a little bit early in the advisory, but I'd rather be a little bit early and get people paying attention than ignore it and have, what we saw in 2019. So we're still fine tuning a number of things about how to sort of deal with the predictive and how do we let people know what's going on.
Thanks, Tracy. Kathy?
So this question is for Dave. I think you mentioned that the Karenia mikimoti cells form cysts in the wintertime to overwinter. Has there been research done to see if the cysts form beds like Alexandrium catenella? Oh, mute. Can you unmute him?
Now,
Pam?
is it okay now?
Yeah.
Okay, sorry. Yes, so these cysts are a little different. So Alexandrium is a thecate dinoflagellate. It has those thick cellulose plates all around it. This is an athecate one. It's essentially a naked cell out in the environment. So these cysts they look like essentially dormant cells without the flagellae. So they're just out bouncing around in the water column, maybe on the surface layer and the nepheloid layer down the bottom. So I don't think there would be the same analogy with cyst beds. It's more there's just a few cells hanging around in the water. When conditions resume, for growth, they germinate and can start another population.
Okay,
So they're a little different.
Thanks.
I see David's busy answering a question from Matt Liebman in the Q&A too. Give him a little kudo for that. Are there other, thanks Kathy for starting that up. Are there any other questions for the speakers from the panelists? Because what we want to do with this is to really come up with a list of next steps. And it sounds like one of the things we need to do is a little more trying to characterize that interface, that area where the blooms are happening, and figuring out what the characteristics are there so that we can predict it a little better and warn people when it might be happening. Leldon?
Yeah, so I, came here expecting to hear there were nutrient-related problems and prepared to explain, what the department is doing in both, surface water and groundwater discharges and how we might direct some of our efforts, to collaborate with the scientists, who presented today and so forth. And obviously that's sort of out the window. So from what we've heard, so I am curious from the speakers if you have thoughts about what role or actions that you might hope to see in the future from regulatory agencies and an agency like myself and EPA, where monitoring and assessment and research is done about water quality. What is it that you would hope to get from not only from these agencies, but from others perhaps that might help you with this issue?
Go ahead, Rocky.
Yeah, okay. I'd just like to say that that one of the challenges of this particular set of events is that we don't even know whether this happened in the past, but we just didn't see it. And so I think that as monitoring techniques improve, develop, continuing the kinds of partnerships that Tracy has put together with the instrument manufacturer, people who it's tricky, as if you get into the offline discussions with Tracy, you'll find out how tricky this is. And but I think that for the DEP and other agencies to try to look for how to effectively develop these industry partnerships to come up with monitoring to push us into these more difficult, where resolution, spatial and temporal resolution are really critical. And it's a little researchy, but it's also incredibly valuable. And so I just think we need to be creative in the ways that we, not just to try to get the shiny object, into our hands, but to really do things in a smart way. And I've been trying to make this argument with MWRA for ages in terms of trying to get more time series measurements. But I think now technology has reached the point where the price point is reasonable to do that. And so this is not I'm not trying to poke Dan Kotiga at all, but if he's listening, that's great.
I'm listening.
Pam, I don't know how to raise my hand electronically.
Oh gosh, yeah, there's a little hand waving at you at the bottom, but I'll put you next after Tracy. How's that?
So I guess I'm going to sort of take this opportunity to make a plug for money, which I feel a little bit cheap and dirty doing. But the study fleet monitoring, the funding that started that was a one-off. So the initial purchase of the equipment and getting that running was a single year source of money, which was fabulous because it let the program start. But what we don't have is a dedicated budget to maintaining the equipment. So the annual maintenance that needs to happen, the calibration pre and post season, the software updates and that sort of thing. And plus any kind of replacement costs for if we were to happen to lose a logger or if one were to fail and, Nick couldn't fix it. So there's, if we want to continue this program, which I strongly suggest that it's a really, really efficient way to get some very high resolution, good quality data with some pretty good spatial coverage. At some point, we're going to have to address the cost question in order to maintain it. And I also think that we'd like to address the gap. So from the maps that I showed, you all could see the coverage that we have, and there's a gap sort of in the center and to the east. And from some anecdotal stuff that I was hearing this summer, it sounded like there was some stuff going on a little bit further to the east than we've seen in the past. And we, DMF put out a couple of our own in-house loggers, and my poor staff went out there and hauled these things by hand, which I owe them all like steak dinners for. But it was less ideal to do it that way. So, we'd like to identify additional vessels and maybe put a sentinel station out. And I'll try to address, Sarah has a question in the comments at the same time, which is relevant. The fishermen right now are putting the loggers in the gear where they want to fish. And they do move their gear in order to continue to maintain their catch rates. And the other thing to mention is that the fleet is relatively territorial. So guys fish in their areas. So there is a little bit of a limit for a particular fisherman to either expand outside of his normal fishing grounds, or it would cost him money to leave the gear in place when his catch rates go down. They have a finite number of traps that they're allowed. So, if they've got a logger in a 10 or a 20 pot trawl, and we ask them to leave that on station, that's a significant amount of their, potential trap hauls that are now kind of wasted because the lobsters have moved on. So that's another thing that we sort of have to figure out how to move forward with. One of the challenges of doing it this way. But I think that that's a pretty lightweight challenge relative to the gain that we get from working with the study fleet. So it's solvable, but it will eventually. We're gonna have to deal with the money question at some point.
Thanks, Tracy. Ken?
Hi. So I'm going to ask a question that's way too big for 8 minutes to 5, but I just want to get it out there for possible future discussions. And like Leldon, I came in expecting people were going to be pointing much more to nitrogen as a potential cause. And I am glad that didn't happen and don't want to try to look for problems that aren't there. But on the other hand, we did establish a warning level for nitrogen for a reason. And just the fact that it's hit a warning level does not mean it's a problem, but it does mean I want to make sure we're really diligent in thinking through every possible way it could be a problem and making sure that it's not before it's time to actually make regulatory decisions in the next round of permitting, for example. So I guess the question I'm just putting on the table is, assuming that all the reasons people have laid out why there's not really an elevation of nitrogen signature in these hypoxia events, assume that's the case. Are there other reasons we should be concerned about the gradual increase in nitrogen levels? Are there other things we should be looking at, running down, making sure we're comfortable that we're not overlooking something before we have to make the next round of decisions? Again, I know that's way too big a question.
And if Karenia gets a hold in one area and then there happens to be some nice nitrogen, some good nutrients nearby that they can move over to. I mean, just feels like less nitrogen, less nutrients is, I'm not going to say blanket, less nutrients is better, but making sure we don't have excess nutrients available is always going to be important. And really, this is one of the things Jeffrey Rosen pointed out, that we really need to have a hypothesis and some questions that we can look at going forward to come up with a monitoring program. Leldon, did you want to jump in there?
Yes, because Ken and I are often simpatico on issues because we deal with so many of the same issues. So I mean, there are plenty of good reasons to continue to address nutrients. They certainly have their own water quality problems within estuarine systems for those discharges to surface waters, and within some of the enclosed and semi-enclosed embayments that we're working on the Cape particularly. I guess, so in response to Ken, I was just sort of contemplating, like, this might be a problem in addition to the nutrient problem, but I am concerned that the nutrient problems and impacts that they have on aquatic life themselves, in concert with sort of a new problem of Corenia, could exacerbate sort of problems within the water bodies altogether. It's an additional problem that might cause additional environmental harm, and so continuing to work on the nitrogen problem would hopefully help to head off a problem of them together.
And going back to something that Mark said earlier about the temperature increases dampening the improvements that we see by reducing nutrients is something we need to pay attention to, because if the temperature is getting warmer, even what little nutrients are there are going to make it easier for the Corenia to take hold. So we're getting to 4:57. Oh my gosh. And I did want to talk about what we're going to do next, because MassBays is committed to making sure we come up with some recommendations. We'll be working with our Science and Technical Advisory Subcommittee, which is part of our management committee, and Presida staffs that and keeps them rolling really well. Dan is the chair, and Todd Callahan from CZM is the vice chair. We're hoping that what we're planning and what we'll do is take the discussion from here, put together a summary that we'll bring to the stack, and ask them to help us come up with some recommendations on next steps that we'll circle back around to everyone here. Are there any final comments that anybody wants to make before we let people go on time? Okay, great. And if there are any, we'll leave the I'll leave the webinar open for a little bit more in case anybody wants to answer some of the remaining questions. And I really thank you all very much for being here on a Friday evening, and have a great weekend.
Thank you.
Thanks, Joyce.
Thank you, Pam.
Thank you.
Yeah, thanks a lot.
Very much.
Thank you, Pam
Thank
and
you.
Presida.
For being here, Leldon, even though we don't get to blame for all the
It's
world of the world.
It's a welcome relief, yeah.
Thank you. Nice job.
Bye.
Thanks, Dan.
Can we save the Q&A?
Yes,
Okay,
I will make sure to do that.
Good job, Pam. Good job, Presida.
thanks.
Have
So
a
we
great
can stop.
weekend.
You
Let me
too.
know if I can help. I'm sure we'll be working on this for a while.
Yes.
Yes, you're
Maybe
you're on the hook there, Dan.
maybe we can stop the recording. Or
Oh Oh yeah, good idea. I can at least stop the recording. Here we go.
Yeah, I can't stop it from